platform. He has over 15 years experience in academia and research. He has done a lot of research in dryland research and drought stress physiology, and has applied enormous techniques in research such as irrigation, fertility, water management and seed systems in ASAL, training and curriculum development, scientific writing and reporting. He has attended several international courses offered by many international organizations such as CENIT, ICRISAT, International Atomic Energy Agency, ILRI, among others. If we come to his educational background, he holds a PhD in crop science with a major in crop physiology and breeding. He has and a master's in crop science and a BSc in agriculture. He has co-authored and authored over 20 papers in refereed journals and attended over 50 conference proceedings in various lines, including crop production, application of biotechnology, and molecular breeding in agriculture, climate change resilience, modeling, among others. He has also supervised more than many postgraduate students, especially masters and PhDs, in local and international university. Because of his good work, he has won several grants in research and development, and has developed many breeds of crops such as beans, chickpea, finger millet, wheat, and sorghum. Some of this work have been funded by international organizations like Gates Foundation, AGRA, and ICRISAT. Allow me to welcome this presenter to give us his keynote address. Thank you. Professor Kimurto, please, you can now, the floor is yours.
I thank the Vice Chancellor, University of Eldoret. She gave us a keynote address yesterday. Colleagues, my seniors, including Professor Ujuodo, Professor Miriam, and all the colleagues in this meeting. I was happy to have been invited to give a talk on a very wide area of topic that when we talked to the organizer, she told me to choose anything around any topic on the areas she gave me. Eventually, I settled on my area of expertise, where I enjoy most. So I focused mainly on climate change. I focused also on areas around crop physiology and breeding. And therefore, ladies and gentlemen, my talk will just be open discussion. It will also include my experience in working around the research area. I've worked also with quite a number of colleagues in this meeting, both supervising students and also doing. So I will just
share your video?
Just give some insights on some few things that I have done.
Sorry for interjection. Could you please share your video?
Yeah, I seem to be. I have shared it. My video is on. I'm not sure whether my camera is not very clear. You can't see me.
We can't see you. Sorry.
You can't see me. Maybe I'm in a network area that is a bit weak. Can I continue, please? I have my video is. Yes, Chair.
Just continue.
Let me continue. So I will spend some few minutes. I have some few slides I have prepared for us to discuss jointly, and I'm going to share now. Yes. So this is the topic I was given. I could not cover everything. At one time, I talked to my colleagues to give us some information on food safety, food laws, food security, and climate change. But eventually, it was going to be long, and maybe in future, Chair, we can also, there are colleagues in Egerton who are expertise in the first area, food safety, food laws, and waste. But I'll mention some few things that they shared with me. So I am a professor in the Faculty of Agriculture. Major area is crop science. And my major focus has been crop science, mainly crop physiology, and interlinked with plant breeding. So I will just give an area I selected, apart from the three areas: research and development in trees to mitigate climate change in drylands for enhanced food and nutritional security. I have case studies right from the pre-breeding into the phenomics and some bit of genomics. How do we link phenomics to improved varieties, and some few examples of the work that we've done with my colleagues. So we know that the agri-food sector in the last 10 years has actually grown parallel to any other dynamics, including trade openness, socioeconomic inequalities. It is a discussion, it's a debate all over agri-food sector. But the threat to food security is still very high. We are told as late as yesterday, somebody passed on in Laikipia because she could not get food after delivery. This is because food security is complex. It's a complex phenomenon that we may be enjoying food here in the highlands, but our brothers and sisters, our Kenyans in the dryland are not getting enough. So we still have very large number of insecure people, especially after the pandemic. Review indicate that from 650, it may go to 800 million people. And it has also increased in Kenya. So in the last UN summit in July, there was a discussion that was led by debates on gaps, opportunities, and barriers in food laws and waste in Kenya, which is highly linked to food security. Because it looks like the waste comes even when we have produced enough in the high producing areas, for example, in Kenya. And maybe this is where the challenge is. And the UN has targeted one of the sustainable goals as food distribution, reducing waste in where there is a lot that is being produced, and trying to create channels and distribution to reach where the insecure people are. And these insecure people are always in the, excuse me, Chair, I've lost the voice a little bit. I'm coming back. I'm back. I think I'm now okay. So the system, which is agriculture, producing the foods we talk about, consume a lot of water. And it also consumes a lot of energy. So it's highly intensive in terms of water, it's highly intensive in terms of energy. And therefore, the energy that could be a lot in the drylands should be tapped to compensate for the less water that is found in the drylands. So the water, energy, food security nexus is very crucial. And it has been actually been picked by the UN as one of the sustainable goals. Because the growing population, rapid economic development and urbanization is actually bringing in demand for water to increase because the diets of the communities are changing. So we may need to be looking at the climate change wiping out genetic resources, and at the same time, looking at which of these require less water, producing more energy, and contributing more to the food security. So these are debates that go on, and we cannot actually eliminate the fact that Kenyan dryland is having a lot of challenges, ranging from poverty. And we know 2 million Kenyans having a problem. The genetic resource species in the drylands are very high, 30%. The mammalian livestock breeds are in the drylands. And both these are facing a lot of agricultural threats. So as we concentrate the productivity in the highlands, the lowlands are having a lot of genetic resources that are being wiped by climate change. For example, the Living Planet Index show that the global population, the wild species, have declined by a very high margin. The tropical species, where we have in Kenya, has gone more by 60%. So as we expand into the traditional, non-traditional production areas, we need to identify and preserve. We need technologies that identify, preserve, and protect these habitats. However, in Kenya, food systems provide food security. It can only provide security when the food supply is stable, affordable, and the prices are determined or stable. But the biggest challenge for all of us in research, of course, this conference is about budget. Yesterday, Kyoko talked about how cereal grain growers are contributing to the food security in the country. But we know that limited budget across the sector is a problem. Because compared to, and this has got very high correlation with our food stability, food supply, food security across the country. Because if we would take 6%, the way Uganda have taken theirs to 10%, if we would take the 6% and put it over our budget and put it in the drylands, which is 80% of our area, I think we may reduce the 2 million people to less than 500,000 in terms of food requirement. So this is one of the factors that we should be talking in every conference, in every meeting, because we know as researchers, we are not getting enough support in terms of national budget. Because Uganda and Kenya, the two economies, the two countries depend on agriculture, the economies depend on agriculture, but Uganda has continuously improved technology in terms of new varieties, storage system, and even off-farm activities are being more supported in all the food, and that's why we see a lot of imports into Kenya. This is something I picked from reading and in the internet. This is why low productivity in the drylands. You can see the determinants of land productivity is storage of water, which is a problem in the dryland because of the nature of soil. The nitrogen and carbon, we don't have the sources of carbon. And we also have limited microorganisms to work on the organic matter, which is also very scarce. And therefore, the carbon, nitrogen, and water scarcity in the dryland makes the dryland have low productivity. So it's an environmental issue, which we need to every factor in this, the metabolites, the animals, the microbes, the water infiltration, the roots, all these, for us to increase productivity, we need to generate technologies. And one of the key things is breeding. The other thing is application of conservation agriculture. The other one is bringing in technologies of agroforestry. The other one is reducing environmental pollution in the water that goes in the drylands, so that we can enhance productivity in this harsh environment. So we need a cyclic resource management in the drylands. If you look at some of the case studies, like in Isholo, there's a case study where our colleagues are working in. They have a project that are looking at the pastoralists as a circular economy. What type of pasture? African foxtail. How is the root characteristics to preserve the soil? How is their dry matter? How is their productivity? How is the organic matter coming from the animals? Combining the livestock so that they don't move to produce organic matter that go into supporting the crop livestock integration. And in the end, reduce fixed carbon, improve or reduce the carbon dioxide that goes into the atmosphere to mitigate against climate change, and enhance the nitrogen cycle. So in these three main components of the dryland, we need a circular system in all of them. A circular carbon, a circular nitrogen, a circular water. All this, Chair, I'll have some few examples of what is happening in the drylands. But we know the drylands have been, the agriculture have been developed. So in the next 20 years, we will see a great and a big difference in terms of agricultural development as per counties. So this is a very interesting picture that is showing our county governments. So our county government must manage the raindrops. This is governance. And when you look at this picture, put it, for example, in Baringo, do we see this? Are we seeing the rain, precipitation, evapotranspiration, infiltration, groundwater flow, water tables, agroforestry, uptake, irrigation losses, for example, in Marigat, how are they being managed? And on the corner here, this is the environmental, we will need weather. For us to manage and to manage every drop of water, we'll need weather forecasting as one of the key tools that in the future to improve the productivity of the dryland, which is improving this, we will need a governance structure with packed mechanisms of enhancing that they monitor for a long period of time the weather patterns and provide information to farmers. The soil is also a problem in the dryland. Three things are happening: the degradation, loss of that photograph, soil quality, and productivity. So there are quite a number of things that can be done. I picked cover cropping, permanent raised beds, so that we have combined, or rather preservation of soil and preservation of water with a target to improve the transpiration efficiency and water use efficiency. This is the volume of water consumed per produce, and also the produce per unit of water consumed, the UST volume of water, and the kilogram of yield per meter square. research from FAO, I checked the research from FAO, their productivity, crop water productivity in the drylands of Turkana is only 25%. The rest of the water go away. So the river Talgwell, the river Kerio, even in Marigat, is slightly better, the water productivity, because of the type of soil is slightly higher, ranging between 45 to 55%, especially in the Verkera irrigation scheme, where they have high value crops like seed and also horticultural crops. So this transpiration efficiency must be increased by enhancing techniques or conservation techniques that are required. And when you see here, on this is a photo of a farmer in Makueni, conserving raised beds and covering with polythene. And this is one of the crops in a rural irrigation scheme. We had a project there some time and we planted this crop. Two things occur. One is when we have proper crop density with raised beds and the irrigation system, transpiration efficiency increases to 70, 80%. Because the yield by unit area, when the surface is covered, the transpiration will be through the leaves. The ET, evapotranspiration, is reduced significantly. That is, evaporation from the soil surface is significantly reduced, and most of the water loss is through transpiration efficiency. So we see more biomass production by volume of water utilized. So in terms of conservation techniques, this is some of the examples that when you look at in the drylands, they have enhanced productivity, and communities and even counties that have adopted conservation technologies have increased productivity. This project was supported by the county, and the farmers increased land area from 1,400 to 2,100 in the current rural irrigation scheme. So there is impact when a technology is introduced in the right way and the farmers are given the right information and the right varieties. These are some of the research findings which on the transpiration efficiency in terms of different crops, and also when you use physiological components to determine the differences in crops and the differences also in the time period. In this table here, estimation of transpiration efficiency, this was a publication by Tan and Sikla in 1983. You actually see that for these three legumes, Phacelia, veg, rye, and mustard, under stress conditions, the veg is more affected for most of them. If you check the transpiration efficiency, the lower it is, the lower the productivity. That means the lower the biomass eventually that is going to be produced. So when you see Phacelia species, it is very high, both under stress conditions and under non-stress conditions. When you look at the value here in grams per millimeter per millimeter compared to the K coefficient, you realize that it is still having the highest. But it tells you that later on for veg, later on without a lot of stress, it also gives very high transpiration efficiency. So this tells you that species have different transpiration efficiency when they are used as cover crops, because this is based on their cover crop transpiration estimates of the FAO model. When you look at the reference crop coefficient, you realize that legumes has got very high, 1.1, as compared to generally legumes as compared to cereal. But when you look at Facilia, for example, the ground cover, veg ground cover is very high. But at the beginning, you realize that the first year, that is 204 for veg, it was low. And then you cultivate the crop, and then come the following year and measure, look at the ground cover, it increases. Similarly here, for Facilia, it is high and it goes down. For veg here, it is low and it's high the following year. What does this mean? It means that once some of the crops respond much more to ground cover as special crops at the beginning, but in the following year, the impact on their organic matter may not be as high to contribute to the productivity of the second year crop. So these are determinants that we can use to select the crops that we can use as cover crops, and the factor of physiology that we can use to select which crops we can use as cover crops and which ones utilize less water when we take them into the dryland areas. This was also one of the studies on the productivity of rice and Clotoria rice rotational system. And you can see that compared to the other fertilizer recommendations, you realize that the contribution of rice yield and the farmer's practice and the 100% recommended fertilizer was very high. But when you have green manuring, the cost is lower, and at the same time, you get almost similar yield with additional benefit of reduced fertilizer poisoning in the farm eventually. This one is on an example of grain yield, same, but conservation tillage, giving you importance of tillage systems, publications from FAO also and Anus et al. In crop science. If you look at rice and the different conventional tillage, zero tillage, residue retention, and then zero tillage, retention with a crop, and then minimum tillage, you see a lot of changes. Being different in terms of Clotoria, linseed, and wheat. When you look at organic matter, the next table, organic matter and earthworm population and the hydrogenase activity, you realize that the organic matter for conventional tillage can never be compared. So there is a link between this and these figures, and even earthworm population, and even the activity of the enzymes breaking down these crop residues. So we may not see the impact when we say, let us do conservation tillage in the drylands, but we will see them in terms of yield, and we also see them when we do microbial analysis. And therefore, Chair, I think this, like soil microbial biomass carbon, this figure here is very, very high for zero tillage, double tillage, and if you also do for the crops themselves, you can see that conventional tillage is actually not recommended in the dryland. When we've talked about the energy, food security, and water nexus. There is some information also on agroforestry, what the soil loss in the agroforestry system, and even the cropping system. If you check the one for farm food forest, it is very, very low, tons per year per annum. Some of them may go The minimum is 19, but this is very low when we have a field of bamboo. So we need to practice watershed, and even in the hanging valleys of the drylands, these are figures from Prasad et al. 1986, and he did this in Brazil. So we have here, when we look at those few examples I've given on data, we have the crop physiology and breeding. And the plant phenomics at the moment is the crop breeding and crop improvement is almost, when you check all the publications around, there is need to combine biology, chemist, physicist, computer, physiologists, genetics to develop new tools and methods to improve plant population. And once these techniques are developed, we can now use to screen large populations for genetic variation up to F2 or F3. And the key biotic and biotic stresses that require multiple genes, difficult to select, like drought, salinity, high temperature, may not actually succeed so much. If you saw the presentations yesterday, it is likely that it took time, and therefore we need to develop the phenotype-genotype interactions, and we need the equipment and facilities, which are actually currently called plant phenomics. And this has been described well, that they should be non-destructive. They should be high throughput, up to 5,000-6,000 generations or populations to be selected. And it should be aiding the marker-assisted selection. This is because the destructive method and manual method are not giving us precision in selecting for good, high-yielding, and stable genotypes for the dryland. So there are 4 tools in the phenomics. 1, resolution images. We also have currently developed the developments in the imaging where there are movement of equipment across a greenhouse or even a field where measurements are done. Powerful the data that is the powerful algorithms for imaging, and also there is high throughput platform, which could be data analysis and even software. So these 3, if you check this photo, this is actually technology application for plant phenomics, and it is a basis of increased plant breeding outputs in the CG system. For us in Kenya and in the National Research System, if you go to Mexico, you see this. If you go to Erie, you go to ICRISAT. This is where we have a combination of data together. The lab sequencing data, genomic data, plus the field data. So this is the image we talked about, fluorescence, thermal imaging, plant height. All these are measured, taken to the data, brought back, and then the genomic data is brought into that. And this is the phenomic platform. It could be in the field, and it could also be in the rain shelter. This data is collected and put together, and it will be very quick to do profiling and to also integrate phenomic data and genomic data. Because if you look at what we do, even in our varieties we developed in Egerton for the beans and the chickpea, it's just conventional breeding. We have introduction, then we have local varieties, we do crosses, we wait for 6 years, we are in NPT in the 7th year, and in the 10th year is when you have a variety. That's why the process has been very slow. Of course, the introduction one is fairly easy because you do adaptability studies, but crosses up to F6, and then selecting the traits, looking at the characteristics. These tools, if we can have them, they are very few in Kenya. like the rhizotome facilities in University of is in KU. Several labs have got this part of the phenomics, the sequencing and molecular labs, but a combination of a breeding platform we still lack in Kenya, where we have all the physiology equipment constructed in one place, all the genetic data, genome-wide association studies done together is still missing. So I think this is one of the things that a chair we could be thinking as one of the facilities that if universities can have facilities or money, they can invest. I've been developing an interest with Professor Kinyua for a long time on roots, and we started with bean box techniques long ago when we were doing our masters. But currently we have root scanning and leaves, a wind rhizome system. So we could be adding here because we have rain shelters, we have wind rhizome equipment that can measure the root length. You can get 10 kilometers of length. One time we were measuring the roots of sorghum, and when we put them on this scanner, it gave us 2.5 kilometers, one variety. So it counts all these roots, it counts all the forks, it calculates the surface area, it counts all the roots from one plant, and it gives you in a computer. Those days we were, this is a box, a bean box technique. You put in a nail here, and then you open the box, wash the roots on the quarter-inch nails, and then you now count. This root goes this way, you count slowly by slowly. If you look at the efficiency of this facility 10 years later, and this one, and then this one, it tells you we really need to do investment. And this is where we were saying, for us to succeed in research and development, we need funding. We have several publications. If you check in the internet, they have several publications that are coming out, both from the labs that have this very difficult area of study called the roots. This phenomics I've of course started almost in around 2000. And you can see here, application of imaging techniques in screening for several crops. For example, thermal imaging has been done in very many crops. You can check these publications. Root architecture I've talked about has been published in rice, has been published in chickpea, it has been published also in wheat. So these are some of the resources we have, which we can use to even do and support our postgraduate teaching and introducing physiology and breeding together. We still have others like chlorophyll fluorescence, photosynthetic efficiency detection. We have the MRI for mostly for those of us who are interested in the root nematode and the root studies and the root entomology and pathology. And we also have those of us who are interested in PET. You can be able to have an array of traits that have been published, and you can use this as a source, as a resource to help us as we even build our facilities. This is one of the studies for my student, where we picked one trait, where we measure the root length density. This is where you use the cylinders, you put in the roots, you can vary the moisture regimes, and at 40 days, you wash off the roots. Because it is known that around 50 days, the roots never grow. They will have reached optimal. So you need to remove them, and you can get genetic variation within the first 40 to 40, the maximum of 50 days. And we published this. You can be able to get, Muriuki is one of my students, and we got very interesting results. Not very high variation in terms of root length density, but we got in the other, like the root length. And we eventually found this variety and this one to be sources of resistance. They had very good trait characteristics as compared to the local varieties, a variety called Gara Loco, and also ICCV 1882. So this, if you wanted to develop population of QTL mapping, you will cross this parent and cross this parent. Susceptible line and a tolerant line for that trait. And you'll be able to do QTL mapping that you can use for marker-assisted pack crossing. There was this publication also on the fababin, comparing the parental material for breeding using phenomics by Renham et al. And they looked at the water stress and the water limited, and you could see a lot of variation on total dry mass. So this is 20% reduction, 35% reduction, 26% reduction, and 14% reduction. When you look at such a graph, you realize that the genotypes are highly varied. They have contrasting response to water stress for this trait. And this is very good for gene mapping and to also get what are the physiological responses and basis for this. So in the end, you discover that a variety like this one, the last one, has got very low responsive to water because the yield reduction is only very, very low, 14%. So we will need to get varieties like this one who give us the highest variation, because this variation may also be related to yield. So in environments where we don't have extreme weather conditions, sometimes you get enough water, such variety can be good for a farmer because he can produce sufficient yield when there is enough rain. When you look at this from Pelachu et al., leaf number, stomatoconductance, and even the photosynthesis, chlorophyll fluorescence, you discover that genotypes vary. And in the end, as you take the traits of interest across the phenomic platform, you should be able to select values with the highest conductance, values with the highest fluorescence, chlorophyll fluorescence, which is for the system 2 productivity when it is under well-watered and under well-watered conditions. In the end, in this publication, it was found that these two genotypes, this one and this one, when you look at their leaf and mass fraction, stem mass fraction, and root fraction, you discover that these were the best because it had resilient traits that didn't change so much under both moisture and stress conditions. So, Chair, we could also give examples of climate smart technologies. This is one of them. Some evaluation of sorghum in Turkana County. When you look at this farmer, this is a logo champlasm, and this is the improved variety. This variation and this tells you that this farmer has planted this seed for a very long time. So crosses and yield reduction is actually the problem. And this is what farmers have. When you have a variety like this, it is fairly red in color, fairly uniform, although it is not, there were seed mixtures. The yield difference in these two varieties will tell you that this farmer will get at least 20 to 25% better yield than this. As also additional traits like protection against bats because of tannins, and also maturity. if you check this, you discover that they were planted at the same time. This will require another 3 weeks to mature. So the climate smart technologies in the ASALs are needed, and some of them are varieties. You could also see this one, a better improved variety, very large panicles, a uniform, planted in the right density, with good biomass, good harvest index, with traits of livestock feeding, because the pastoralists will require crop residues for their farms. This shows that we can have climate smart crops that can keep farmers up to more than 50% of their varieties they have. However, these varieties must be farmer preferred, because these varieties may not be as tasty. We discovered that in Turkana, this variety can be eaten whole. You eat the stem, you cook the grain, and you also have this as sugar, because children can feed on this when it is still young. These heads can be boiled as compared to the conventional. This is Ekaton sorghum one, which is very good for food. So this tells you that we need to combine these traits and these traits to get some level of acceptance by the farmer, increase the yield, and be able to give them the farmer preferred varieties. We saw some, of course, yesterday, even Kipno presented resilience. He presented differences in the yield of dolichos in his study. And one of the things that was very, very important was to check whether the varieties had resilience to diseases and pests, which are very serious in the drylands, sensitivity to the farmer preference in terms of color of the crop or the grain. And I think it was discussed that this is an area that in the drylands, we can actually be able to improve the farmer's productivity. This is the same photo. And there is also one of the projects that we've done also in that region on groundnuts. And this is exploitation of the resources and the crops that have never been introduced in these areas. And it is very easy to convert the food insecurity in the region into a better productive area if we can introduce these groundnuts and grow them under the irrigation scheme. We found one of the interesting things in this region as management of resources and governance and communal organization is key for uptake of technology. When you see, this is 5 acres, and these women are supposed to be organized to irrigate this area together. If you ask yourself, who will organize them? There is need for a strong governance in some of the irrigation schemes in Kenya. And one of the problems that has led to the breakdown and loss of the irrigation schemes that were developed by the Red Cross in along the river, the Kerio Valley, is because of governance. There is no way you will increase the ease of new uptake of technology through governance and utilization of resources. So this is one of the examples of governance in area of uptake of resources in the drylands. Extension training also is key, because if you look at this variety, this is groundnut, this is peanut. We know it produces flowers at the top and it goes into the soil to produce a seed. Many of the extension staff in these areas may not have this information. So training them is key, and including them even developing a good seed system, which involves both the community seed system and also the formal seed system. This is one of the biggest bottlenecks, and it was mentioned yesterday, that as we take crops into these areas, we need to have a well-established seed system. The mechanization also is key because women and these foods are together. There's a lot of tragedy. The food producers are the women, and many men need to be brought back to the field through motorized handheld equipment. This is one of the irrigation schemes where FAO supported the introduction of IL equipment, as both you can see they can plow, they can transport, and they are able to actually improve the efficiency of the productivity in the drylands. Chair, I think the food loss is also a key thing in the food systems and food waste. There are two different things. One is intentional and the other one is not intentional. So for those of us who are in production, we talk about food loss. For those of us who are in the consumption side, we call it food waste. And even in our homes, we are actually experiencing a lot of food waste. So these are intentional discarding, and the other one is a production loss, accidental loss of quality and quantity. And so many studies, and we are all doing research around this area, and I earlier indicated that food loss and waste would lead to more efficient land. If we can reduce them, we can actually impact on climate change and livelihoods. Because the numbers are there. The numbers are here. In Kenya, we are wasting 99 kilos of food per year. So we throw away 5.2 tons of this, including the milk we saw sometime in Nyandarua, is because of the lack of facilities and the infrastructure to keep the food. So in the groundnut value chain, the critical areas of protecting losses are listed here. We've done a survey and we found that we have several areas up to storage, critical points of yellows, and within the yellows, we have also introduction of aflatoxin. So the shelling using hand machines as compared to shelling using hands, and the health conditions of the farmers in Turkana, for example, and even in the drylands of Kenya, makes our food not go into international market. Because the introduction of, we don't have toilets, we are handling food, both at production and at a transportation level. The health status, the health standards of our food is of course mismanaged from the farmers level all the way to the market. And therefore, we did some aflatoxin analysis along the Baringo, Geomarakwet, Turkana. We found that they are very low parts per billion, less than 4 parts per billion at harvesting. But when you come to Eldoret, or you go to Nakuru, or you go to Kisumu, and do an analysis of the aflatoxin contamination at that point, it is more than 10%. Why? Because the environmental conditions at harvesting and selling around December is that the relative humidity of the environment does not allow Aspergillus species to grow on the ground. But when we introduce it in the markets and we begin to handle it, that is when the aflatoxin gets introduced, and the numbers go beyond the European Union levels. So I think there is need to do processing at farm level, and then transporting the farm produce without the market, without handling along the value chain. Aggregation should be done in the farms and delivered directly to the factories. Because in Turkana, for example, the Jandaria industries are the takers of the peanut, and they are the ones that are supplying food for the refugees, the 400,000 refugees in Kakuma. And they import 6 million kilos of groundnuts from Argentina every year, which is billions of money. Process it in at the river, transport it to Turkana as Nutri-Food to feed the malnourished children. The area in Turkana is very adequate and ideal around Baringo, and the whole of the North Rift, the valley is ideal for groundnut production. The link between the industry and the consumers, even at that level of international organization in Kenya, is also very weak. However, the contamination is key. It may not meet the standards if we don't come in with the technologies to avoid handling of the produce by the farmers who don't have toilets. So I think, Chair, as I finish, these are some of the examples of contamination during drying, during shelling, and even insects themselves create a lot of losses when they attack the crops. So we need the pig packs, even for our own homes. And the impacts of food losses is 30%. Impacts of food losses and waste is 30%. So every 70 kilos is available, even if we produce, the rest go into food waste. So your topic and your selection of the area as a discussion in your conference is very important. I want to close it there, Chair, and thank you and thank everybody for listening. I may not have gone into the details of our research much more because I found that it was going to be too long. We can discuss. Thank you, Chair, and thank you again for inviting me to do this talk.
Thank you so much, Prof, for that wonderful presentation, taking us through the production factors and all through to the food loss. At this point, I'm going to welcome the audience to ask one or two questions based on the things they were not able to capture while you are presenting, or any clarifications that they may need. So the floor is open. Maybe as the other members are thinking of presenting, I just have one question for you. There's a slide you are presenting on the impacts of agroforestry on transpiration and so on, and the data seems to have been captured in 1986. So do you think the impacts could differ now, because that is like nearly 30 years ago, and you realize that there are a lot of things that change, and even the climate patterns and so many things change, especially when you look at production. Do you think you would get the same results now?
Hey, Chair, I think I was expecting that question. When you look at that transpiration efficiency study, it was one of the most referred. And even if you read the current publications, it is being referred to that, because you may get slight changes in the values of K coefficient, crop coefficient, especially when you integrate cover crops and agroforestry. The values you get may not be varied to any significant level. So I decided to use that as a base, because it is a very good study that is well published in a very good journal. There are many others that have been published thereafter, including the ones that have been done by our colleagues in Baringo, Cameroon. We have that data. So I think you are right. but not much change in terms of the values, especially the physiological values of crop coefficients and crop factors. I hope that agrees with you, Chair.
Thank you, thank you. So I'm going to welcome the floor to any member who wants to maybe ask a question. So just raise your hands, then I can give you the chance to. Yes, Dr. Were, please go ahead.
Thank you, Emmanuel, for giving me this opportunity. I wish to highly commend the presenter, Professor Kimurto, for giving us a very well-researched, very enlightening presentation, accompanied with very good results that have been published. I have found this presentation very interesting, and I'm particularly touched by that bit that was about soil degradation and the procedure that involves raised soil, and the pictures that was shown in Marigat of a farmer who had raised the soil and the farm in Makueni. I found that very interesting. I'm just wondering, Prof, can this procedure be applied elsewhere, or is it specific to dry lands? Can this procedure be done on other crops? Can it be done on other crops?
Yes.
Or is it specific to those crops that were shown?
No, it can be done anywhere. You could see several publications in rice. It is also there. You only get optimal transpiration efficiency when you apply more in a less resource environment. The idea is to ask ourselves, do we raise beds for maize in Uasin Gishu or in Nakuru? Do we raise beds for wheat in the highlands? If we raise the beds, we may interfere or even don't get the efficiency in terms of mechanization and other related activities. We have this study, and even the technology becomes much more efficient in water scarce environment, because you are using flood irrigation. And if you go to Marigat or even Aror or the whole of the north, even parts of Isholo, the farmers get irrigation water on few number of days. And they may be getting that irrigation water for few number of days in a month or a week. So raise the beds, and the crops are indicated are actually an indication that you may not apply in a different environment. It would work very well in areas where land is flat and where the water needs to be optimally used. I've not done myself a study on the TE efficiency under these raised beds. We only did a production. That those crops you saw was a production. It was not a research.
Okay.
I hope Gertrude, you are okay. I hope it's convincing.
Yes, thank you.
Yeah.
Yes, it is. Thank you.
Okay. Thank you, yeah. Thank you, Prof. I'm going to invite Professor Chudo to also ask a question, then Dr. Amy.
Thank you, Chair. Thank you, Paul. It was nice
Thanks, Julius.
Hearing you and listening to your wealth of experience. Nice that you accepted to interact with us. I have two issues might be comments. In them, there could be questions. One is financing research and financing universities. You have commented and very well that for us to do very good research, we need proper financing so that we can have the right infrastructure and the right personnel, because there is need for a lot of collaboration. And you went into showing us how people are moving towards looking at below the ground characteristics of crops to determine tolerance or even varietal differences. And you made mention of very expensive equipment that we require. Is it your view that we should have these equipment as universities? And if that is your view, do you think every university should be able to get or to have such equipment? I'm asking this because we know that our universities rely on government for most of our capitation, and that source, as you know very well, is drying up very fast. So what can you suggest to be the way forward? And even as you answer that, I would tell you that other places, what they have done, I went to school in South Africa, and they impressed me in one thing. During apartheid, South Africa had 9 state universities, 1 in each of the provinces. And because they know that finance is not adequate, they planned, the government planned that each university could have 1 important equipment that would be shared by all the universities. I don't know whether that is something that you can recommend here, but you have your experience. Maybe you can tell us. That is one area: finance, infrastructure and equipment, and the way to go in our research. The other one is, you talked at length about climate smart agriculture, conservation agriculture in the drylands. But I want also to bring in the constraints that we experience here in the fact that most of our farmers are small scale. Is conservation agriculture applicable to our small scale farmers? And more so when you look at our small scale and they want to plant all crops, how do we go about this? And more so with the view that we have today, that we want to encourage our farmers to embrace commercial farming, where they must organize themselves properly, they must have properly organized seed systems, they must adopt mechanization, etc. How do you see this in our situation? Be it dryland farming or small-scale farmers, and your idea of conservation agriculture, climate smart agriculture, and the way our farmers are. Those are my comments and questions. Maybe you can respond. Thank you.
Chair, I could start from Ojuoda is thinking very deep about our problems in universities, and even us at Egerton. We are still on strike, negotiating with lecturers now to start. But let me start from the last part of it. Conservation, yes, conservation agriculture. I think you have a point. you're a very valid point. Small scale and the technologies required. My suggestion, and I've seen some improvement on this, we may not succeed on some areas of conservation agriculture. That includes an example, technologies around mechanization in small scale farms through conservation agriculture. The implements required for conservation agriculture are not yet in the market for small scale farmers. At the moment, when you look around, we are only seeing motorized equipment to support reduced drudgery, plow faster, weed faster, spray, weeders, sprayers, transport, and plows, motorized. And I gave that example there. It's an old one. We can succeed. In fact, this is my experience. We can succeed in conservation agriculture among the small scale farmers on crop-based technologies, cover crops, cropping systems, crop diversification, agroforestry. Those are the ones that we can promote, and they make sense. They make sense. But because, for example, I gave an example. We want to conserve water after harvesting. We want to conserve short rain water in the drylands. We have grown sorghum and we have harvested. That sorghum has extracted so much of water, and it has also extracted so much of microorganisms, of the nutrients. If we want to apply conservation agriculture, we must bring in a crop that will act as cover crop. It will act as a source of legume, and it will leave 10% of the crop residues, because the farmers will harvest everything and feed their livestock. If you grow beans, if you grow green grams, if you bring something that can help to conserve or utilize the water. We talked about crop productivity and economic water productivity. So crop water productivity is an additional output from a liter, a millimeter of water. If we say we put in a conservation, crop-based conservation technology, we will plant crotoria, for example, or the one that we gave, or even plant canola or plant any crop. Even if the farmer, small-scale farmer, will want to feed his livestock with the crop residues after harvesting that crop, there is 10 to 15% natural vegetation drop. Natural. There will be a natural that will remain in the farm after harvesting that cover crop. The large-scale farmers will come in, like the Nightingale in Nakuru, will come in and plow sunflower after harvesting into his farm. He will come and plow in the green peas, the green manure. But the small-scale farmers will never do that. I'm one of them. Just near your door or just next to your door, there is a goat, there is a cow, there is a sheep, and the land is so small. So we have to actually be very realistic, Prof, that for me, the experience is it can only work for now for the crop-based technologies. The large-scale mechanization, the big things we talk about, implements that cut crops, straws, all that will not work for now. The other bit, I hope that can. So 50%. When you go to more irrigation scheme, you realize that as compared to aero, you realize that, and also when you look at irrigation schemes in the Elgeyo Marakwet and in Turkana and Baringo, immediately after harvesting maize, all the irrigation schemes are open for grazing. In Mwea, they don't open for grazing after harvesting rice. They have a second and a third crop. They have a ratoon and they have a legume. So those are the areas where the conservation agriculture technologies can work. In other areas, the livestock will just come and clean everything and expose the field, eat everything. There will be more or less than 1% crop residue coverage. The equipment and the universities. You are very right. And we need to start this because we will be forced to do this shortly. We will be forced naturally by the need to naturally contribute more, not the basic things that we are doing. The standards of training, research in Kenya is going up with very less resources. We are fighting for a strike in Egerton now. We have a lab that was built by Chinese. We got 80 million molecular lab. It has most of the things, but the utilization is very low, less than 50%. We don't have students who are in plant breeding. We don't have enough professors who are in molecular genetics. We are shy to go and run a PCR. We don't have money to equip the lab to have all the chemicals. So, and I'm sure I visited my colleague who is having a Striker isotone facility in KU to do for my student to do some. We were doing some Striker studies there. The guy, yes, he's called Professor, I'm forgetting his name. He has a very good facility for phenotyping Striker. It's not anywhere. I've never seen it in East Africa. I think it could be the only one where you can detect the pheromones, the compounds that are released by Striker, the stimulants can be analyzed in that facility. The facility is 30% use because KU don't have a lot of agricultural programs like Chepkoilel or Egerton or Nairobi. So, Prof, I think the future is we must start collaborating. The equipment are going to be available, in my view, in some few stations, in Kari, in Egerton, some few equipment, and we need to go the university, the South African way. We have two problems. One, money to buy the chemicals. Even your lab, the molecular lab, I normally come there. It's the same. Number two, the equipment are not fully utilized. Number three, we don't have enough capacity to use them. If you put all the students from Egerton and Chepkoilel for plant breeding, it can be taught by all of us who are in that area, plant breeding and physiology. And this is for a fact. If you put the special postgraduate programs for Chepkoilel and Egerton, we can actually come together as a group and teach them at Egerton on practicals and teach them at Chepkoilel at some given time. So I think that's my view, and it's a good question. I may not have answered them well, and we may be having different opinions as academia in this, but I think that we will be forced to go there. Thank you, Chair. Sorry, I've explained a lot, but it makes a lot of sense to share our opinions.
Thank you, Prof, for the feedback. Professor Chuda, are you okay with the comments?
I am okay.
Okay, thank you, Chair.
I hope we are agreeing.
Dr. Ines just said that.
Julius, I hope we are agreeing.
Has been captured by Professor Chuda. I just have a question on aflatoxin. I actually am impressed that you brought the issue of aflatoxin and you are explaining the reasons why maybe it is prevalence in one geographic region and not the other. Do you think that other mycotoxin producing genera like penicillium and maybe fusarium could also exhibit the same mechanisms like that, climate would be a big factor in explaining why it's prevalent in one region and not the other.
I think so. Although we've not done any studies on this, the chair, but I think so. It's a relative humidity. It's an RH factor. So although aspergillus is more resilient, and we did a comparison between even in their control using the aflasafe plus other species of trichoderma, we are seeing we may eventually get more high efficacy molecules and the microorganisms. But I agree with you, it's a relative humidity factor. So we need to think around that in terms of reducing the aflatoxin levels.
Thank you for the feedback.
Can I add my voice to that, please?
Right.
On aflatoxins?
Yeah.
Yeah, it is true that the levels of aflatoxins depend on the crop and depend on the storage conditions. More so, the moisture content of the grain and the environmental vapor pressures, meaning how much moisture there is in the atmosphere under which the grain is stored. And that is why you find that most of the research in this country have pointed out that there is more aflatoxin in the western part of the country and the eastern part of the country, especially the Ukambani areas. There's a lot of research that has already come up from this, from people in Nairobi and several other places. I can tell you for a fact that as early as 1985, when I did my master's was about aflatoxin in maize grains. And I collected maize grains from all over the country. And the only grains that had aflatoxin at a level that was not acceptable was grains from Meru and Ukambani area. The rest of the country had some level of contamination, but it was relatively low. This has been confirmed by other people like Mudomi, Nairobi, and the rest. And even here recently, we have had students who have looked at aflatoxins in foods. In the School of Environment, there have been 2 PhDs there. In our school, both in crops and animal science. So aflatoxin is a big issue around, and it is a storage issue that needs to be handled. But there's a lot of work that has already gone through. Thank you.
Thank you so much, Professor Kimurto and Professor Chodo for that. If there's no other question, then I would again request us that we clap again for our keynote speaker for that insightful presentation.
Thank you so much. Thank you, Chair.
So from here, we are going to have some break. Then we will break into the rooms after the break. So the program might delay a little bit, but we are going to just have a small break, tea break, then we can resume into the various room sessions. Thank you for attending, and I encourage you to be patient and also attend the rooms based on what you would want to hear. Thank you so much.
The University of Eldoret is a modern university situated approximately 9 kilometers northeast of Eldoret town in Uasin Gishu County, Kenya. Founded in 1946 and converted to a teachers training college in 1984, in 1990 it became Chepkoilel campus of Moi University, offering natural, basic and applied science programs. In August 2010, the campus was upgraded to Chepkoilel University College, a constituent college of Moi University. Upon the award of charter by the President on 11th February 2013, the University College was renamed University of Eldoret. Our vision is a premier university that nurtures global leaders and innovators. Our mission is to provide quality education, training, research and consultancy in science, agriculture and technology to meet the needs and aspirations of a dynamic society. The University of Eldoret is ISO 9001:2015 Quality Management System certified. The university is led by the Chancellor Ambassador Professor Judith Mbula Bahemuka, who is the titular head of the university, the council comprising of the chairperson and members, and the University Management Board led by the Vice Chancellor Professor Teresa Akenga. The other members comprise of Deputy Vice Chancellor Academic and Student Affairs Professor Ruth Otunga, Deputy Vice Chancellor Administration and Finance Professor Wilson Ngetich, Deputy Vice Chancellor Planning, Research and Extension Professor Philip Raburu, the Registrar Academics Professor Paul Taruz, Acting Registrar Administration CPA Francis Ikapal, Registrar Planning Dr. Benjamin Mwasi, Acting Finance Officer CPA Ereu Totona, and Senior Legal Officer Mr. Stephen Anditi. University of Eldoret is science, agriculture and technology based and has grown steadily from the initial schools of forestry and science to the current 9 schools with 34 academic departments. The schools are led by a team of deans who also provide academic leadership and management of the faculty. They include Dr. Oliver Kiplagat, Acting Dean, School of Agriculture and Biotechnology, Professor Robert Otuya, Dean, School of Business and Management Sciences, Dr. Paul Odwori, Dean, School of Economics, Professor Kisilu Kitainge, Dean, School of Education, Dr. Julius Kolongei, Dean, School of Engineering, Professor Gelas Simiyu, Dean, School of Environmental Studies, Dr. Jacob Onyango, Acting Dean, School of Human Resource Development, Professor Boaz Kaunda Arara, Dean, School of Natural Resource Management, Professor Samuel Luta, Dean, School of Science, and the Dean of Students, Dr. Lelei Kiboi.
This is one of the big schools in the University of Eldoret with several programs covering all the fields in agriculture, both at undergraduate level and postgraduate level. Also, we have some diploma programs. We strive to be a center of excellence in agriculture, as our vision is to be a center of excellence locally and globally in agricultural sciences, technology development, and transfer. We have a total of 6 departments within the school, which are under the dean. So we have a wide range of courses, and our students are not limited to work in agriculture, although most of them work in agriculture, but we have other options which are not traditionally agricultural enterprises, but nowadays employ our students.
The School of Education is the largest school at the University of Eldoret. We serve about 50% of the total student population. The school is housed at the main campus in the education complex and also at the town campus located at Musco Towers opposite Sirikwa Hotel, Eldoret town. The School of Education is organized in 8 departments. We at the School of Education prepare teachers and trainers to be employed by the Teachers Service Commission and the Public Service for Technical and Vocational Education teachers. Our graduates go into industry and private practice. The School of Education programs are highly popular due to the many avenues for employment after undertaking the courses. Graduates can go into teaching, they can go into industry for technology education, and also some establish their own businesses.
The School of Engineering. The School of Engineering was founded in 2011 with 3 departments which are fully accredited by Engineers Board of Kenya. The first department is Agricultural and Biosystems Engineering. The second department is Civil and Structural Engineering. And the third department is Mechanical and Production Engineering. All the above 3 programs ran for 5 years. The School of Engineering has actually a number of unique strengths. The first one is that we have qualified and dedicated staff with varied experiences in different fields of engineering. Secondly, our programs are marketable, both at graduate and undergraduate level. We also have excellent teamwork among academic and technical support staff and also with students. We also have good cooperation and networks with neighboring institutions, industries, and other stakeholders. Without also forgetting, we are also located in a serene environment next to a fast-growing city. The School of Engineering has 4 administrative units. Mainly, apart from the 3 departments I have mentioned, we also have the school, the Dean's Office.
Professor Samuel Luther, Dean, School of Science, and an Associate Professor of Chemistry. We have a mission and vision. Our vision is to be a vibrant center in excellence in generation of scientific knowledge. And our mission is to create, innovate, disseminate scientific information through quality teaching and research. As a school, we have 4 departments, namely Department of Chemistry and Biochemistry, Department of Physics, Department of Mathematics and Computer Science, and Department of Biological Sciences. We offer programs both in pure and applied sciences, and our programs are market-driven and sensitive to global trends in terms of innovation.
Jerusalema ikayalami ilondoloze uhambe nami zungangishilana. Jerusalema ikayalami ilondoloze uhambe nami zungangishilana. Dawo yami ayikholana, buso wami awukholana. Ilondoloze zuhambe nami. Dawo yami ayikholana, buso wami awukholana. Ilondoloze dawo yami ayikholana, buso wami awukholana. Ilondoloze zuhambe nami. Dawo yami ayikholana, buso wami awukholana. Ilondoloze zuhambe nami. Lema ikayalami ilonoloze uhambe nami zungangishilana dawo yami ayikholana buso wami awukholana ilondoloze zuhambe nami.
The University of Eldoret is a modern university situated approximately 9 kilometers northeast of Eldoret town in Uasin Gishu County, Kenya. Founded in 1946 and converted to a teachers training college in 1984, in 1990 it became Chepkoilel campus of Moi University, offering natural, basic and applied science programs. In August 2010, the campus was upgraded to Chepkoilel University College, a constituent college of Moi University. Upon the award of charter by the President on 11th February 2013, the University College was renamed University of Eldoret. Our vision is a premier university that nurtures global leaders and innovators. Our mission is to provide quality education, training, research and consultancy in science, agriculture and technology to meet the needs and aspirations of a dynamic society. The University of Eldoret is ISO 9001:2015 Quality Management System certified. The university is led by the Chancellor Ambassador Professor Judith Mbula Bahemuka, who is the titular head of the university, the council comprising of the chairperson and members, and the University Management Board led by the Vice Chancellor Professor Teresa Akenga. The other members comprise of Deputy Vice Chancellor Academic and Student Affairs Professor Ruth Otunga, Deputy Vice Chancellor Administration and Finance Professor Wilson Ngetich, Deputy Vice Chancellor Planning, Research and Extension Professor Philip Raburu, the Registrar Academics Professor Paul Taruz, Acting Registrar Administration CPA Francis Ikapal, Registrar Planning Dr. Benjamin Mwasi, Acting Finance Officer CPA Ereu Totona, and Senior Legal Officer Mr. Stephen Anditi. University of Eldoret is science, agriculture and technology based and has grown steadily from the initial schools of forestry and science to the current 9 schools with 34 academic departments. The schools are led by a team of deans who also provide academic leadership and management of the faculty. They include Dr. Oliver Kiplagat, Acting Dean, School of Agriculture and Biotechnology. Professor Robert Otuya, Dean, School of Business and Management Sciences. Dr. Paul Odwori, Dean, School of Economics. Professor Kisilu Kitainge, Dean, School of Education. Dr. Julius Kolongei, Dean, School of Engineering. Professor Gelas Simiyu, Dean, School of Environmental Studies. Dr. Jacob Onyango, Acting Dean, School of Human Resource Development. Professor Boaz Kaunda Arara, Dean, School of Natural Resource Management. Professor Samuel Luta, Dean, School of Science. And the Dean of Students, Dr. Lelei Kiboi.
This is one of the big schools in the University of Eldoret, with several programs covering all the fields in agriculture, both at undergraduate level and postgraduate level. Also, we have some diploma programs. We strive to be a center of excellence in agriculture, as our vision is to be a center of excellence locally and globally in agricultural sciences, technology development, and transfer. We have a total of 6 departments within the school, which are under the dean. So we have a wide range of courses, and our students are not limited to work in agriculture, although most of them work in agriculture, but we have other options which are not traditionally agricultural enterprises, but nowadays employ our students.
The School of Education is the largest school at the University of Eldoret. We serve about 50% of the total student population. The school is housed at the main campus in the education complex and also at the town campus located at Musco Towers opposite Sirikwa Hotel, Eldoret town. The School of Education is organized in 8 departments. We at the School of Education prepare teachers and trainers to be employed by the Teachers Service Commission and the Public Service for Technical and Vocational Education teachers. Our graduates go into industry and private practice. The School of Education programs are highly popular due to the many avenues for employment after undertaking the courses. Graduates can go into teaching, they can go into industry for technology education, and also some establish their own businesses.
School of Engineering. The School of Engineering was founded in 2011 with 3 departments which are fully accredited by Engineers Board of Kenya. The first department is Agricultural and Biosystems Engineering. The second department is Civil and Structural Engineering. And the third department is Mechanical and Production Engineering. All the above 3 programs ran for 5 years. The School of Engineering has actually a number of unique strengths. The first one is that we have qualified and dedicated staff with varied experiences in different fields of engineering. Secondly, our programs are marketable, both at graduate and undergraduate level. We also have excellent teamwork among academic and technical support staff and also with students. We also have good cooperation and networks with neighboring institutions, industries, and other stakeholders. Without also forgetting, we are also located in a serene environment next to a fast-growing city. The School of Engineering has 4 administrative units. Mainly, apart from the 3 departments I have mentioned, we also have the school, the Dean's Office.
I am Professor Samuel Luther, Dean School of Science, and an Associate Professor of Chemistry. We have a mission and vision. Our vision is to be a vibrant center in excellence in generation of scientific knowledge. And our mission is to create, innovate, disseminate scientific information through quality teaching and research. As a school, we have 4 departments, namely Department of Chemistry and Biochemistry, Department of Physics, Department of Mathematics and Computer Science, and Department of Biological Sciences. We offer programs both in pure and applied sciences, and our programs are market-driven and sensitive to global trends in terms of innovation.
university has directors who coordinate the formulation, review, and implementation of university policies. They include Dr. Victor Kimeli, Directorate of Information and Communication Technology; Professor Donald Otieno, Director Quality Assurance; Dr. David Waswa, Director Strategic Planning and Performance Contracting; Professor Beatrice Were, Directorate of Board of Postgraduate Studies; Professor Vincent Sudoi, Director Research and Innovation; Professor Grace Cheserek, Directorate of Industrial Linkages, Partnerships and Collaborations; Professor James Olekiapi, Director Resource, Mobilization and Strategic Initiatives; Dr. Geraldine K. Motola, Director Gender Equity and Diversity; Dr. Nicholas Rope, Acting Director, University of Eldoret, Eldoret Town Campus; Dr. Nicholas Macau, Acting Director, Open, Distance and E-Learning; and Engineer Emmanuel Kinyor Mutai, Acting Director, Income Generation Units.
We were later informed that it begins at 11. and make losses or cause losses in our farms. So we have seen that actually these chemicals have got quite a lot of challenges. And the only way out of it is biological control. And in this case, I'm looking at the use of entomopathogenic fungi. Back, please. I'm looking at the use of entomopathogenic fungi to control these insects in our farms, even in the greenhouse, because they have the advantage. They do not need to be ingested, like the nematodes.
Julius, you are on and off. You are on and off.
Really? Sorry, I don't know what is the problem.
You maybe are moving around.
I'm not moving.
Okay, focus onto the microphone.
I'm seated. So what I'm saying is that entomopathogenic fungi have got several advantages.
Yes, that is fine now.
Compared to other entomopathogens, which are viruses and also nematodes. One of the advantages is that they would directly penetrate through the cuticle. They don't need to be eaten or to be ingested by the insect to cause infection. Also, after they have been sprayed in a field, they overwinter in the soil, because most of them are isolated from the soil. They overwinter and they can cause infection in the subsequent seasons. The other thing is that they would reduce or decimate insects pest, and at the same time, some of them act as endophytes, and I've got some interactions which are also beneficial to the plant. Next, please. So you can look at this picture. This is the entomopathogenic fungi mode of action. I think what I have just sent has not been, it's not what is being shared. Kindly someone should check, because I have got some very interesting information in that as I continue. So in this work, I did some classification of entomopathogenic fungi. These are all over the world, and you can see in the phylum Ascomycota, the many that we have. Then we have some in Basidiomata, not so many. Then Entomophthalomycotina are also having several of them. You can see a lot of entomophthorales are actually entomopathogens or entomopathogenic to insects. Go to the next one. Then we have Chidiomycota, and some are in Zygomycota, which are not presented in this table. They have several of them that are also pathogenic to insects. Next, please. So if you look at this, we have got several insects that have been infected by several fungi. We have the orders Lepidoptera, Hymenoptera, Diptera. In fact, several of them, almost all orders, can get infected by these fungi. And we have different types of species of fungi that you can see. We have Gibellula. We have got Beauveria, which is very common. We have Cordyceps. We have Isaria. Go to the next place. Metarhizium is known to most of us, I know. Hypocrella, which is on the far left, you can see some yellowish. It's on a scale insect. And you can see the last one on the far right, and the last one, which is as actually costomycosis on Lepidoptera. You can see acanthomiasis. So that was just to give you an overview of how these fungi are very critical and they can really be useful in biological control. These are some of the insects that we collected in several parts of Thailand, in the northern part of Thailand. Some of them we collected from the forest. We collected when we were trying to isolate some of the fungi. Next, please. So I told you that today I will try to focus more on Africa, but this paper covered both Africa and Southeast Asia. So you can access the paper later, or I can share it if you need, where you will now get more information. But the pictures that I have just shared or talked about, the pictures that I've talked about are in another paper, which I will also share with you. It is on a book chapter, which we also published with my colleagues. Now, in Africa, the first that I would have noted that was produced or used to produce a mycoinsecticide was metarhizium in South Africa, when there was an invasion of locusts. And the mycoinsecticide, or rather the biopesticide that was made, was known as green mussel. That I'll put a picture there from Kabi. You can see someone is spraying the green mussel, and this is on maize. You can see it is on maize. And during that time, there were really devastating maize in South Africa. And this green mussel has been known to be good in both locusts and grasshoppers. Now, move to the next one. Next slide, please. So in Kenya, I'm just giving a brief on this. In Kenya, we've had quite a number that have been isolated and used to control ticks in their carry in cattle. And you can see this is metarhizium anisoplea being sprayed. That's why you can see the green, because it causes the green muscardine. It's being sprayed on a bull that has got a lot of ticks, and thereafter you can see that the tick has been being colonized. Then in Tanzania, there was a successful use of Aspergillus oryzae to control tuber absoluta, and within 3 days, there was 70% mortality. Next, please. In Ethiopia, they have used both Beauveria and metarhizium to control termites. These termites are very dangerous, and also in Kenya, they are very dangerous. They would cause a lot of damage to wood and timber. At least I've seen this in my area. But you can see by the 3rd day, there was 100% mortality when they used Beauveria and metarhizium. I have also seen metarhizium being used on termites also in Asia, and they have been very, very successful. Next, please. When you go to West Africa, Benin, used Beauveria against mosquito. And in this case, it was not really about mortality. What happened was that it caused, it altered their blood-sucking behavior, and this reduced their blood-sucking behavior. So even if they would get a host, they would not suck as before. And in Nigeria, we have examples also in Nigeria, and also a very interesting one in Malawi, where Beauveria bassiana was found as an endophyte in sugarcane, and also at the same time, it played a role in controlling some insects in sugarcane. So next, please. Wow, what has that happened? There had some data that I wanted to show you. Can you change the person who is can you give me the other presentation that you removed, please? The one who is the other one, the PowerPoint, the PDF, the PDF.
Okay, this is the one that you sent the latest. Let me check the PDF.
So what I wanted to tell you is that in Africa also did quite a lot of search, and realized that we have developed quite a number of biopesticides that are useful in agriculture. And I'm happy to report that Kenya is among the best in Africa that is doing quite a lot of work in this. And I think thanks to ISIPE, and is followed by South Africa in both adoption and application of entomopathogenic fungi in insect control. And this is very good for us because, you know, now people are more keen on good food, or rather food that is not having a lot of chemicals. I also realize that when you want to export your produce to countries like Europe and the US, they want zero chemicals in them. And this gives us an advantage. No. This is a presentation I made yesterday, not this one. The other one that you are giving us first. Not this, please. So in Nigeria, they have also done quite some. But the problem that we have in Africa is that we have only dwelt on 2 types of entomopathogenic fungi. That is Beauveria bassiana, of course, various isolates or various, and Metarhizium anisople. However, we have got a lot that have been isolated, but they have never been, and they have been isolated and tried and seen to work, but they have never been used to produce biopesticides. And that is what I would want to encourage us on, that we should do more, that we should do more research and focus on how to make them available to the farmer so that they can be useful. You could see in the classification there, we had got several entomopathogenic fungi. I know some are difficult to grow, some are difficult to culture, but for the ones that can be cultured, we can use them, because you realize that most of them are specific to various insect pests. So we cannot use Boveda bassiana to control all insects, because for you to get a very efficacious one, in most cases you have to isolate it from insect, or when you go to collect them, you will get them from insects. So when you get a particular one from a particular insect and you are able to culture that and make it into a biopesticide, that would be more efficient. So I think I would just encourage us that we can also read further on this paper. You will find it online. And the other one, there's another paper which I've not indicated here. I can share both. I don't know how to share a document here.
Thank you, Julius. I think you better conclude, because
I've concluded.
We've taken a lot of time.
I've concluded.
You've concluded.
Yes.
Thank you very much. I think this is an interesting area. It's a very rare area of research. If you can share those papers of yours later on, I think that will be very beneficial to the rest of us. Thank you very much. I don't know whether anybody would be having a question to put to you. Anyone? As Mr. Oberlin gets ready. Any question? Oberlin, you'll be ready. So you are the next. So be ready with the Bambara nuts. Any question?
I think maybe I was very audible and everyone listened well.
It is all right, Julius. Your area was nice, despite the fact that you did not have the data. The presentation was okay. Yes. Yes. Now we will come to her last, because she was not available when she should have been available. Gerald, you are ready?
I'm ready, Prof.
Okay, let's see whether these guys of mine can upload the document for you. Please share it. It's coming. Okay, Gerald, there you are. Please, another 15 minutes.
Okay.
Yes, Gerald, go on, please.
Yeah, can it be on full presentation mode? Good morning, ladies and gentlemen. I'm Gerard Obalim. I'm a PhD candidate at the Seed, Crop and Horticultural Science Department.
It doesn't change. Okay, Gerald, it is not changing here, so maybe you'll just go on.
Okay. Yeah, I want to talk about Bambara nut, which is a neglected and underutilized legume in many parts of Africa, including Uganda. But some work that we have done shows that it has some promise for resilience of these communities who are growing Bambara nut, especially in the dryland regions of Uganda. Next, please.
Move the documents. Move it.
We are moving.
Okay, sorry for the technicalities. Give us 1 second to sort out the problem.
That's okay.
You should open it in the presentation mode. Okay, is that okay, Gerald? No?
Yeah, that's
Move up
okay.
again.
Give us that first, that slide. The very first slide.
Move up.
Yeah, that slide. Next, familiarize ourselves with what we want to know. We know about this crop. It's yes. To ground. Okay. Can we move to the next, please?
Move to the next.
Gerald, you are breaking. Kindly find a stable connection. You are breaking.
I'm sorry, madam. I think that is going to be a bit of a challenge for me. I thought this was the best spot I could pick.
Okay, Gerald, momentarily, I think you can switch off your video. It will a bit improve your network.
Okay.
All right, go on, please.
Yeah, the video is off.
Yes, now we can hear you well.
Okay.
Good.
Yeah, so it's generally just a subsistence crop. And like I mentioned earlier, it's grown mostly in the dryland regions, but yields have been reported in elevations up to as high as 1,600 meters above sea level. So that is still another area that is quite interesting to explore. Among the legumes in Africa, the food legumes, it is the third most important. That is, of course, after groundnuts and cowpea. That tells you it has some standing in among that group. And it's a very nutrient-dense legume. It is high in carbohydrate and protein and has some good quantity of fat. So it's a nutritious legume. And the filling effect normally reported about it is, I think, because of that high carbohydrate content. Other attributes like drought tolerance, performance on poor soils, and providing a good intercrop for other climate resilient crops such as millet, sorghum, and cassava makes it a very good prospect, really, because we are at a time where climate change related issues such as prolonged droughts, loss of genetic resources, dwindling food sources, all of these are threatening food security. And if you have a crop that is very drought tolerant, a crop that can fix nitrogen, a crop that requires minimal resources for cultivation, and our farmers are into that kind of a system, I think it makes Bambaranat a very good strategic crop moving forward as far as resilience and sustainable agriculture and climate change adaptation is concerned. So this is why we are interested in exploring more about this crop. But the problem is that in Uganda and arguably in most other parts of Africa, this crop has very little information that has been consolidated about it. In Uganda, it's actually almost non-existent. We don't have production and utilization information. And this was one of the first of its kind of study that we conducted. So we needed to go ahead to find out production and utilization practices and identify the challenges and opportunities that exist for the improvement of this very promising crop. We did a baseline survey in June, July 2019. We considered 12 districts in northern and eastern region, The reason we did that for those two regions is we purposely sampled those two regions because we were reliably informed by technocrats in those areas that those were the predominant production areas. They lie in the dry areas of Uganda. There are other dry zones, but they don't produce as much as these other two regions that we chose. And we assigned districts proportionately, still based on reported levels of production. Then we chose districts within each region and sub-counties within each district randomly. And the respondents, we used two methods of selection. We purposely selected only Bambara nut farmers, and then we also randomly selected from within the Bambara nut farmers, those that were available at the time of the study. We looked at a total of 371 households, and we interviewed them face to face. We gathered information on demographics, scale of production, gender roles in production and utilization, and then the challenges of Bambara production and utilization. And the percentages that you see here, we have generated them by descriptive analysis among all those factors that we considered. Next, please. This picture is a briefing session during the survey. We had to first get our respondents together, and then we brief them on what kind of work we have come to do, getting their consent, et cetera, and then of course they knew about their crops, so we didn't have to tell them anything about their crop. So that was a session in I think Parabongo in Agago district. That's okay. Now what we found out was quite interesting. We saw that there was some marginal increase in production levels, what was harvested and what was sold. This quite highly correlated. This information is in another manuscript which has been submitted to a peer-reviewed journal already. The increase was in a period of 3 years. Much as that doesn't extend very far, but it told us something about what trend is coming up as far as that crop is concerned. I would like to bring to the attention of my audience here that these two regions had disruption in agriculture for quite some time because of civil unrest, and crops like Bambara nut almost disappeared. And when we saw that trend coming back, we felt this was something interesting because that means people are slowly returning to growing this crop. Related to that was the number of people who were not growing the crop. That decreased in the same period. And related to that, of all those who were not growing the crop, who had grown it before, 97% of them were willing to come back and start growing this crop because they had issues that they want to address as far as that crop is concerned, such as food security, nutrition, etc. So they showed a high level of willingness to re-engage in production. We felt that was some good indicator of why that crop holds value to them. These all scenarios, they all suggest a steady increase in the area of Bambara nut that will be produced in the subsequent years. So this can be consolidated by a younger generation, which is surprising for a crop of that value, a purely subsistence crop. We found that 21 to 40 years of age, youth in that category, were very much involved, almost just like the elder ones, beyond 40 and all that. So that tells you that there's a future there, that if these people are interested in doing that at that age, they will probably still be interested in the next 5, 10 years, etc. And another interesting thing which we found was that these farmers seem to know a lot about the quality of the seed they want. Things to do with seed selection, threshing. They told us they thresh seeds differently from how they thresh that when they want to prepare for food. They store seeds differently from how they store that one for food. So they kind of have that quality assurance at many stages in the seed cycle, which was confirmed by laboratory tests. We picked some seed samples and analyzed them in the lab. That is presented in a publication that Obura and other friends worked on, including myself. So good seed quality, quality assurance of seeds makes it interesting in that these guys know what they want, and they are intent on doing what is the best to keep going. This picture tells us of a field in Kapelebyong district in eastern Uganda. It was the largest field that we recorded throughout the study. It was about 3 acres. What that means is that there is will and there is motivation out there. We just need to find that and encourage it, because this guy knows what he's doing. He's why he's growing a lot of that, and that means there's hope for other farmers to also benefit in the same way. Next, please. Now, another aspect of resilience that we noticed was the symmetric nature of gender roles in production and utilization. When we talk of the symmetry here, we mean the similarity in roles between men and women and the youth. I would like to draw attention to the different colors of the bar. The pink are the ladies and the blue are the gentlemen. As you go to the right from seed to marketing, you see the roles of men diminishing and vice versa. And the other interesting thing I want us to draw attention to is the green bars, which of men and women. What that tells us is that there's nearly equal involvement between the two gender in as far as activities of Bambaranat are concerned. Seed means seed selection. I think there's a caption down there that explains a bit of those. So what that means is women and men share roles, and the men involved are also equally willing to do most of the things that are needed for the crop. And this, we go next, please. This is manifested in both the eastern and the northern region. Things are similar, although it's slight variations here and there, but the same message that we were able to see from here is that men and women share roles in Bambaranat production and utilization. Next, please. This is a portrayal of the total picture, I mean, the overall picture from all the respondents. And it is exactly what we've just been saying, that the role sharing is something that kind of comes out as unique when you consider a neglected and underutilized and a subsistence crop. Next, please.
2 more minutes, Gerald.
Okay, Prof. These were some of the reasons they cited. Traditional practice, that meant it is just what they've been used to doing, that it is something in the community that you do it as a man or you do it as a woman. And then some would say it's just about the knowledge about the crop that makes you take those decisions to be involved in those activities. Some of them say it is the household head decision. He takes it upon himself to make the decision on those activities of Bambaranat. Next, please. So this symmetry of roles is different from what is known about separation of roles in food crop and cash crop production, whereby most food crops are for the women and cash crops are for the men. That situation is not the case of Bambaranat in Uganda. And what that means is that it provides a platform for wider participation of the communities, that more people can actually be engaged in Bambaranat, given that you would only expect women and elderly people to do that. Now we looked at some of the challenges these people are facing. The main ones were these seed scarcity, limited market, harvesting, and storage. The disclaimer about limited market here was that the market was there for fresh boiled snacks within the communities and all that, but not for bulk sale of grains and seeds. So the market is there, but people are willing to have more. They want to get more money out of it because they normally do piecemeal harvesting, and when they harvest in bulk, they fail to find the bulk market for those. So those are some of the major challenges that we were given as major obstacles. However, next, please. Most of these challenges seem not to be overburdening the farmers, because they seem to find a way around most of them. Like when it comes to seed scarcity, they normally save their own seeds, or they are normally given by neighbor, or they're given by somebody, a relative or someone, okay? And instead of limited market, I mean, in terms of limited market, what they normally do is they just like to consume it fresh boiled or sell in the local market so that they don't bother too much with looking for the bulk market. And they always plant in smaller quantities as well, okay? So that they can offload all the products. The same thing applies to difficulty in drying. I mentioned briefly about seed quality assurance. This is detailed in Obura's publication. They can dry longer period, they can dry on tarpaulins, they can also do redrying for storage pests and diseases. So as far as tackling these challenges are concerned, this is not unique to Uganda. These challenges exist elsewhere in Africa. And the way the farmers deal with them, they're just almost exactly the same as what we found in Uganda. And this is because Bambara nut is neglected, it is underutilized. There's not a lot of research work going into it. So what farmers do is deal with the problems the way they know best. Either knowledge has been transferred to them or what they have learned from experience and other crops. But what we want to say here is that the persistence of farmers in trying to solve these problems and then continue to grow Bambara nut, for us, it points to a sign of resilience and of sustainable farming. Now, beyond these farming communities, we have a lot of other opportunities. There's a lot of research that can be done on Bambara nut at all levels: agronomy, entomology, pathology, etc., etc. There's still so much to be done about Bambara nut, and that will all give us more information that we can use to promote the crop. In terms of germplasm, we managed to collect a few, but that wasn't rigorous enough. We still believe there are a lot more other materials outside there, which research can try to look at and then bring them together. IHEA has a collection, but I don't think they have all of that from Uganda. I think Uganda can still contribute more materials. I may be wrong about that, but I'm quite sure, because when we went to the field, there were a lot other things that was going on in terms of the variability in the seeds. Now, in terms of seed production, this is the part which is kind of easy to tackle, because there's a model which has been promoted by the integrated seed sector development called the local seed business model. And this has the partnership of a research institution with local seed businesses that are existent in the community to produce quality seeds. They call them quality declared seeds. I think Bambara nut would really easily fit into this model. This would help them with the problem of scarcity of seed. And then community need to know that time is changing, the weather is changing, climate is changing. We will soon go back to the food we used to survive on, and Bambara nut will be one of that. So they need to be educated about such things. Now, we conclude that production and utilization of Bambara nut in Uganda has shown some good attributes of resilience for communities and for sustainable agriculture. And we need to push forward with that, because most research agenda is about food security, and then the smaller crops or the lesser known crops are neglected, but they contribute to that so-called food security and sustainable agriculture. So we recommend that production practices can be improved, maybe not from the standpoint of research as such, but from the local level, whereby farmers are given basic information about, spacing, etc., and later crop management. And then the research linkages I've already talked about, and these will enable availability of seeds, and all that will help us to increase production and make better utilization of Bambara nut in Uganda. Hopefully that will galvanize the resilience and sustainability of the communities within where Bambara nut is always grown. We are the last slide, please. This work is under SCIFSA, and it is funded by the Inter-Africa Academic Mobility Scheme of the European Union stroke African Union. We are thankful to our mentors at the University of Eldoret and my parent institution, NARO, for granting me study leave. Thank you very much for your asante sana. Poe mata.
Thank you very much, Gerald. I don't know whether somebody has a quick question or we leave question to the end. Anyone with a question as Mr. Bote prepares himself? Gerald, how do you propose that we will be able to increase productivity of this crop? Because it is one of the indigenous crops. It is neglected. It is grown in very small areas. Then we keep on telling ourselves that indigenous crops are cheap to grow, so we don't use fertilizer, we don't do many things. How do you suggest that we can go about improving on productivity of this crop?
Okay, thank you, Prof. I think the first thing is what I hinted on, the issue of their availability of seed. Many of the respondents were telling us that they're willing to engage action when seed become available. And this seed issue is because of the factors that I highlighted before. Some of them lost their seeds, some of them just abandoned farming altogether, and now they're just returning to it. So let's first start by having some seed available. And these seeds can be the preferred types. That means we'll have to go backward a bit and say, which types do you guys like? And then the local seed businesses and the research stations can just multiply some specific types and quantities of seed that is available. And we mentioned that the farmers that are involved, both the men and the women, they're both willing to take it up at all stages, which means the ones who are there are willing to be there. And it makes it a bit easy to work with them because they know exactly what they're looking for, and they know that their excuse will now not be seeds. So we first make the seeds available, and then later on, we start looking at improving other production practices.
Okay, thank you very much. Thank you. I think we'll allow the others maybe to ask their questions later on. So can we have Mr. Bote on, please? Thank you very much, Gerald.
You're welcome, Prof.
Okay. Yes, Hilary.
Hello, Prof.
The field is yours.
Okay. Yeah. Thank you. Good morning.
Morning
I
here,
think
yes.
Estelle, yeah, morning over there too.
That's okay.
Yeah. Thank you for the opportunity. I want to share some aspect of my PhD work. Good morning, everyone. I'm Hilary Mrekubote, a PhD candidate also with UoE, the seed department, and I'm presenting on improved seed quality of African eggplant through after ripening by in-fruit post-harvest storage and fruit maturity. Next slide, please. For those who are not familiar with this crop, it is more grown in West Africa and also some parts of Central Africa. This is the Gilo type of the African eggplant, Solanum aethiopicum. And you could see the top one is the plants bearing the fruits in the field. And the two cultivars that I worked with are Pando and Oforiwa, and the right side is the seed. Please go on. Yeah, so as a small background and also the problem that I sought to answer, the Gilo group is the third most cultivated and consumed in Ghana, and like I mentioned, other West African countries. This crop is nutritionally dense with a lot of antioxidant vitamins and minerals, which are very good in terms of providing the needed nutrition for people in SSA. But the major problem currently in Ghana, and most part of this is that it's still considered underutilized in terms of its commercial production. And so farmers often rely on informal seed sources. Currently, there's no seed supply system, which is being formalized. And therefore, farmers may harvest fruits for the fresh market purposely, because that is what they harvest for the market to sell. And unfortunately, these precocious harvests have rendered very low or low germination percentages, exhibiting some erratic germination behavior, which affect nursery seedling establishment. And so the objective was to evaluate how the right harvesting maturity and the technique, which is called after ripening, would affect the seed qualitative and quantitative germination behavior of this crop. So in terms of research question, there are two that we are seeking to answer. Whether the seeds within the harvested fruits continue to mature, because if they continue to mature, then it means that the germination capacity and behavior is likely to improve. And the second one is whether this technique called after ripening can influence the other physiological behavior. Yes, next please. Yeah, so this was conducted in the UoE experimental research field between July 2019 and February 2020. As I mentioned, the cultivars that I used were Oforiwa and Pando, which have different fruit shapes. So there was a nursery stage, then a field experiment. So the maturity stage that I employed were from 30, 40, 50, 60, and 70 days after anthesis. The DAA is days, the number of days after anthesis or the flower opening. And the storage period that which is the after ripening technique I employed was from 0, that is the same day extraction, or I stored it for 5, 10, and 15 days under ambient condition of an average 24 plus or minus 2 degrees Celsius. And so after each of these storage conditions or maturity stages, the seed physiological quality were assessed. Next, please. Okay, so by way of presenting the data, many physiological quality measurements were taken, but for the purpose of this presentation, I will limit myself to the few of them. Data on thread and dry seed weight were taken. A seed moisture content was taken, and the 1,000 seed weight was taken, as well as the germination and vigor measurements. The current slide that you see as the figure is the effect of the after ripening on seed moisture content. And as you could see, from both cultivars, the moisture differences at harvest, that is the day 0, was very high, ranging between 79 to 80%. And this is a typical behavior.
Yeah, off. Hillary, can you hear me?
Yes, I was muted, I think, by accident. Can you hear me now?
Yes, I can.
Okay. Yeah, so maybe just to go back a little, on this first slide is the seed moisture content at different maturity stages as affected by the after ripening technique. And like I was saying, in both cultivars, there was a more or less a similar pattern in terms of decline of the seed moisture content over time, over the storage time. But what it could be said is that the moisture content decreasing pattern was more of a function of the maturity. That is, as the seed matures, the seed declines because there is an inverse relationship with the dry matter accumulation. And so as the seed matures, the seed is filled with dry matter and food reserve, which is needed for the development of the seedling and later. In terms of the after ripening technique on seed moisture, this one was minimal, though significant, but it was more significant when seeds were harvested earlier, within 40 to 50 days than at the later stages. This is so because at the later stages of 70 to 60 days after anthesis to 70 days after anthesis, this is where the seed attains physiological maturity as reported by Bote et al. In an earlier study. Next slide, please. Yeah, so what I observed again for this slide and the next slide was presenting how it also affected first germination count, which in seed science is a symbol of seed vigor. So you could see for this very cultivar Ophoriwa, the maturity stages and how they were influenced over storage period for the first germination count and germination has been presented. As you could see, there were no germination when seeds were precociously harvested at 30 days after anthesis. And it was also very minimal when they were harvested at 40 days after anthesis for the first germination count. But when they were stored up to 15 days in storage under ambient condition, there were marginal increases in terms of the seed vigor from 0 to 27%. And as you could see from the far end of the germination percentage, this one was relatively improved. So what could be said is that the seed within the harvested fruit, even when detached from the mother plant, continues to mature and improve in terms of its germination capacity. So the present resource is consistent with other reports that have been reported by Bote and also Vidga for pepper, that seeds are able to, seeds that are born in fleshy fruits, such as the African eggplant, accumulates dry matter even when it has been detached from the mother plant. That is why we are seeing some improvement in germination of their seeds. Next slide, please. Yeah, so this is the same for the other cultivar, which behave almost the same. But what should be noted is at 60 and 70 DAA, there were quite improved germination in terms of vigor, first germination count vigor at 87% and 92. And this is so because from other studies, this has been identified as the stage where the seed is physiologically matured. And physiologically matured, it means that it has the highest maximum dry matter and also the highest germination vigor percentage. Please, next slide. Yeah, in measuring the germination behavior of seeds, it is not always okay to just look at percentage germination and vigor. These days, seed scientists have been encouraged to look at other germination behavior patterns which consolidate the overall germination behavior. And so I look at mean germination time and time to reach 50%, which are very critical for nursery practices and also planting purposes. The mean germination behavior tells you the number of days the seed lot requires to complete its germination cycle. And so you realize that as the seed matures, the time required to complete its germination reduces. So from 10 days to 7 days in both, and for the from 9.7 days to 5.6 days. That means that as the seed matures, it has an inverse relationship with the mean germination time. And this is so partly because as it matures, then it accumulates enough food reserve that gives the seed initial energy and a boost to improve its germination. In terms of the effect of after ripening technique or post-storage technique, it was also highly significant. When seeds were not stored, it took more or less a higher number of days to complete its germination cycle, but it reduces as seeds were stored up to 15 days of storage. And these have been reported consistently with other reports for Wilbur and Bradford in the 90s, because at this stage, the seed is known to be behaving, having an in situ priming principle. That means while the seed is in the fruit, it is being primed. And so the moment you release it and dry it for germination, it takes that kind of energy out within the soil to improve its germination. Yes, next slide, please, as I'm trying to wrap up. Yeah, so the other measurement, quantitative measurement of the seed physiology that was taken is also the GI, as the germination index and mean daily germination. The GI combines both the speed at which the seed is germinating and also the overall germination performance, which is also a good measure to tell you the germination behavior and pattern of the seed. And this was also, so the higher the germination index, the more vigorous or germinable your seed lot is. So you could see that when seeds were stored for 15 days, the germination index was higher, reaching up to 4.85 in seeds that were harvested at 70 days after anthesis. The mean daily germination, on the other hand, tells you how many seeds germinate per day, averagely. And so that could also be inferred from the results that the more your seed matures and the longer you keep in after ripening or storage, the number that has the ability to germinate increases up to 7 averagely per day. And this is also trying to consolidate the overall germination improvement that you see in seed lot when you apply the after ripening technique on your seeds that have been harvested earlier. Next slide, please, by way of conclusion. Yeah, so what has been concluded from this study, though other aspects have been taken out from this presentation, but I can briefly say conclusively that seeds of African eggplant within the fruit continue to fail and mature. That has been observed in a relative crop, that is the Solanum melangena, what is popularly grown and eaten in East Africa, that is the aubergine, and other parts of Asia. So even when you harvest the fruits from and detach it from the mother plant before it attains physiological maturity, the seeds within still continue to mature. But when you precautiously harvest the seed at 30 days after anthesis, regardless of you subjecting it to post-harvest storage techniques or after ripening techniques, the physiological quality will still not improve. And this is so because it has been observed that at this stage, the seed has not attained the essential characteristics with a well-developed embryo that contains enough food reserve for the purposes of germination. So, and the last conclusive statement is that the after ripening treatment up to 15 days is able to improve both the seed vigor and also the germination quality, especially when the fruits are harvested at 40 days to 50 days after anthesis. I will take this opportunity to acknowledge our sponsors, that is KEFSA, funded by the EU community, and my institute, the CSIR Crops Research, Ghana, where I'm back working, and also the University of Eldoret, especially my supervisors, Prof. Ochodo and Dr. Ngode. Thank you very much, Medasi Asante-san. Questions are welcome.
Medasi. Thank you. Thank you very much, Hilary. That was a very clear presentation from our two guests from outside Kenya. Any questions from the audience? We have some 2 minutes. As Mami Sodu prepares herself, Mami Diop, prepare yourself. Any questions? So Hilary, this is a crop that the fruits mature at different times.
Yes, please.
You have red and you have some that are still green or yellow. So for seed purposes, what do you advise the farmers to do?
Yeah, so for the purposes of seed, we advise farmers to harvest for the cultivar Ophoriwa, which is the round cultivar. We advise them to harvest when the seed is 60 days after flowering. But in place of that, there is also a color relationship because some farmers will not, in terms of, practically be able to count or calculate the number of days. So when the seed has attained the orange color, vivid orange color, then they can harvest it for the purposes of seed. But for the market, they can harvest at 30 days after anthesis when the fruit is still cream yellowish in color.
Okay, thank you very much. Anyone with a question? Thank you. Maybe we'll have a short while after for a few more questions. So thank you very much, Hilary. Mami, are you ready? I'm not seeing her on. Is she on?
Yes.
Oh, ça va?
Good morning, everyone.
Morning, Mami. Go on.
I'm Mami Swadujo. Today I'm presenting one part of my thesis on seed quality of velvet bean in Western Kenya. Next, please. Mukuna Puyans, commonly known as velvet bean, is an annual climbing legumes. This leguminous crop is endemic in India and other tropical regions, and it is native to tropical Asia and Africa. Just like other beans, for example, soybeans, common beans, it is grown under moist and warm conditions and in both cultivated and wild form. Velvet bean can be used as human food, livestock food, and has medicinal values. It also improves soil fertility through nitrogen fixation. It can contribute to sustainable agriculture and crop production improvement. Despite its multiple use, farmers in Bungoma County are facing problems due to its poor germination and crop establishment in the field. Farmers in Western Kenya face a number of food production constraints like land degradation, low soil fertility, poor rainfall, climate variability resulting in high food insecurity. Adoption of legumes with high protein content such as velvet bean can reduce food insecurity in Kenya. Farmers in Bungoma County were actively involved in velvet bean production due to its multiple use. It can be used to improve soil fertility, root tolerance. Variation in seed characteristics has been reported to be useful for crop improvement program. However, production of quality seed was a challenge for farmers in this county. The purpose of this research was to investigate the quality of velvet bean seed. Seed obtained from a survey in Bungoma County were taken to the seed physiology laboratory, and experiments were set up to determine the quality of velvet bean seed in Bungoma County. Next, please. Seeds were characterized into different types, and seed coat color, ilium color, seed length, width, and thickness was evaluated in 4 replications of 50 seeds, and electrical conductivity was determined in 3 replications of 50 seeds. Next, please. 3 replications of 50 seeds from the 4 types of velvet bean was counted and placed into their respective container in order to determine their vigor. Seeds were incubated for 14 days while monitoring germination count daily according to ISTA. Next, please. In order to know the optimal measure for water uptake of velvet bean seed, 2 measures were used, sand and filter paper. Water imbibing capacity of seed was calculated according to this formula. Next, please. This is the data analysis. Next, please. Next, please. Among seed types, there was significant difference in all seed thread measured except seed coat thickness. White and brown seed type had significantly longer and wider seed than other seed types. Next, please. Correlation of seed thread showed that seed length correlated with seed width and seed thickness. Seed width and seed thickness also showed a significant positive correlation. Seed length and width positively correlated with germination percentage. Next, please. Using the Royal Horticultural Society color chart, seed type were identified. Velvet bean seed coat ranged from black to dark grayish, yellowish brown to yellowish gray. Also, dark grayish, yellowish brown, and yellowish gray brown spots were observed respectively on the seed coat. Pinkish gray pattern color was also on the seed coat of the white seed type. Next, please. All seed type had a yellow-white ilium color, but the group different from yellow-white to white. Next, please. The present study indicated that the variation in velvet bean seed morphology is manifested mainly in seed size, thickness, color, and spot ornamentation. White and brown seed type had significant longer width seed than other seed type. The difference was not significant between black and dark brown black seed type, also between white and brown white. This observation can be explained by the interpretation of both genetic and varying environmental conditions. Correlation of seed ratio is that seed length correlation with seed width and thickness. Seed length and width positively correlated with germination. This is in line with studies that have indicated that big seed goes hand in hand with seed variability and bigger. The role of genetic and environmental conditions in seed morphology is also supported by the correlation between seed threads. Seed threads are important for identification of species. This is in line with some studies which indicated that seed characters are very helpful for identification of large number of species or genera. Next, please. The seed color vary from black to dark grayish yellow brown to yellowish gray. Four color of seed were recognized, and the four seed types corresponded respectively to 4 varieties of velvet bean in Madagascar regarding to color of tegument. However, 7 different exceptions of velvet bean were identified by Guru, Monti, and al. Next, please. All seed type had a yellowish white ilium color, but the group different from yellowish white to white. Pattern and ilium color are also used for space identification. Studies indicated that seed coat pattern or micro-ornamentation on the surface of outer cell wall can be considered of taxonomy value in the identification of species. Some study also indicated that the structure and morphology of the seed coat can be used as taxonomy feature. Next, please. There was no significant difference in the average electrical conductivity among seed type. However, white seed type with average electrical of 0.0012 was more vigorous than dark brown black, followed by black and brown white seed type. The lower the electrical conductivity is, the seed is greater the seed vigor. Next, please. The result is in agreement with Mavi, who indicated that in watermelon, the electrical conductivity decreases from light colored to dark colored seed. There are factors which influence the EC values such as initial water content, temperature, time of seed soaking, the number of seed per sample, seed size, and genotype. Conductivity tests can be used to predict the field emergence and standard germination. This is consistent with studies which indicated that the EC test has been proven as indicator of vigor in wide range of seed crop. Final germination percentage and speed of germination index significantly differed with seed type. White seed type had significantly higher FGP and SGI than other seed type. Next, please. Seed color play a role in seed dormancy and germination. The result observed in black and dark brown affirms that with Shashalis, who indicated that light colored seed type absorb water rapidly than dark soybean seed and consequently have better rate of embryonisation and germination. This present study is in agreement with Bashuado and Modi, who indicated that the color receipt of some legumes, the lower germination was attributed to the tight adherence of the seed pod to the ambient and the presence of phenolic compound. Next, please. Seed structure can be affected by different seed color resulting from various levels of pigment accumulation. This can be explained by the fact that different seed color exerts differential germination restriction action by providing different level of impermeability to water, oxygen, or the mechanical resistance to radical construction. In our study, the lower germination of black seed could be attributed to the greater amount of pigmentation that solidifies the seed pod and increase its impermeability to water and oxygen. Next, please. Water imbibing capacity of the seed significantly different among seed type. As temperature increases, the water imbibition capacity also increases. The same observation was indicated for 3 sorghum species. However, the alternative temperature, 30 to 20 degrees Celsius, resulted in completely interruption of the water uptake for both media. In all 2 cases, the seed revelated the lowest water imbibing capacity at ambient conditions. The maximum ambient was reached at 20 degrees Celsius. Next, please. This is in line with King and Kinder, who indicated that several legumes species, the reduction of imbibition rates is correlated with seed pod pigmentation. This was demonstrated by honey locust seed, whose swelling percentage was increases more rapidly in yellow coated seed than in light and dark brown seed. Similar results were found using soybean seed for seed with black seed pod. Next, please. Also, the ilium can prevent water uptake by acting as an hygroscopic valve. It is known that imbibition capacity is important for germination. This is consistent with Ashraf and Fuller, who indicated that during the imbibition process, seeds are irradiated to a moisture level sufficient to initiate the early event of germination, but not sufficient to permit radiate prostitution. Germination starts with imbibition, a process of water uptake by the seed caused by different water concentration between seed and environment. Next, please. In conclusion, we can say there was a wide variety of valuable seed grown by farmers. These seed types differ morphologically each other in parameters like seed coat, seed color, seed size, and germination and vigor. Dormancy could have a factor that limited the germination of dark colored seed. To increase in production of value seed, selection criteria need to be adopted by farmers, and the barrier to germination in dark colored seed need to be addressed. Next, please. There is a need to standardize agronomic and seed management practice of value in order to improve seed quality and yield at farm level. Genetic diversity exists among the evaluated seed of value. Genetic characterization of value found in Western food seed type have low, yes.
Okay, go on.
Have low germination percentage compared to other seed types. Further study can be done on genetic and physiological property of velvet black seed type in order to improve its capacity. Next, please. I wanted to thank my supervisor, Dr. Mena and Dr. Prof. Oshuado, Victoria and Gigi, Dr. Ruth and Mary. Also, special thanks go to the University of Eldoret and ZIFSA project. Thank you so much.
Thank you very much, Mami. That was a lot of information on velvet beans. I'm hoping that people have learned much about this crop. Any questions from the participants? Any question to Mami? Mami, you're saying that the black seeded types have low
Yes.
germination.
Yes.
And you're saying that something should be done to them. What do you mean? What can be done to them to improve their performance?
Maybe you can look at about the genetic aspect of this black seed type and also the physiological property in order to improve its germination capacity. Because I have done a dormancy breaking test, but also the black seed type failed to have higher germination. But we can say that according to my field experiment, when the black seed type was grown in Eldoret, we have a higher germination, but it is a little bit less than what is recommended. So maybe we can look at about the genetic aspect and the physiological aspect to see how we can improve its germination. Also breeding, maybe breeding aspect can be looked at.
Okay. Thank you. Any question? If not, then we have Brian, and after that we'll have Anne. So Brian, are you ready?
Yes, Prof, I'm ready.
Okay. There you are.
Okay. Is it known already? My name is Chila Brian. I'm presenting on enhancing access and utilization of quality sorghum seeds for improved food security in West Kenya. My co-presenter is Prof. Julius Ochuado. Next. Okay, the background information. Hunger and multiple malnutrition threatens millions of people in sub-Saharan Africa, whereby 30% of the population is malnutrition. As of 2019, 234 million people in Africa were chronically malnutrition than in any other region. In the Africa.
Hello. Brian, where have you gone to? Hello. Amepotea. Brian, are you in a matatu or what? Hello. Hmm? Hello. Hello. Okay, we seem to be having a problem with Brian and his microphone. Anne, are you there? Hello. Anne, are you on? Yes, I am. Are you hearing us? Yes. You are saying I am. Hello. Anne, are you on? Hello. Anne, is your micro? Are you hearing us? Yes, I can hear you. But we cannot hear you. Is Brian on? Brian, are you on? Hello? Anne, have you come in? Hello? Brian? Anne? Brian? Brian? Anne? Hmm? They can use their phones. Hello? Hello? Brian? Hello?
You're the only one.
Anne? Anne? Hello? I can see you are back, but hello? Hello? Anne? Anne, you have logged in again, but you cannot be heard. But if we cannot hear you, then how are you? You are hearing me on my phone, or are you hearing me when I talk on the? But we cannot hear you.
Can you use your phone to log into the meeting? I'll just share your work. You just present. Can you log into using your phone? Yeah. Can you do that? We cannot hear you at all. Huh? Can you log in using your phone? Yes, using your phone. You can just present with your phone. I'll show you.
Hello. Hello.
Brian.
Hello. Yes, Prof. Hello. Yes.
Where did you go to?
I don't know why you are not getting me. I've decided to use my phone.
You've decided to use your phone?
Yes.
Okay, that is fine then.
Okay.
So let's bring back your presentation.
Okay.
Mnaona vile nyinyi mnasumbuana.
Okay, I use in the background information.
Yes, now move very fast because tell us important things kwa sababu hakuna time.
Okay, I was dealing with sorghum. Sorghum is a drought-resistant crop and an integral part of diet. And it is used by, it is recommended for patients suffering from diabetes and HIV. So it's an important crop. However, there's been constraints such as lack of quality seeds, access of quality seeds, as most seed companies shy away from producing this crop. Let's go to the next slide. Okay, my problem was the lack of access of quality seeds in sorghum, and this leads to low production because seed companies do not produce this type of seeds. So farmers mostly obtain their own farm saved seeds. Therefore, because the crop is important, there is need to come up with a good seed system that will increase the supply of quality sorghum seeds. Next. Sorghum is an important crop, and its utilization is closely related to maize. So it can be an alternative to maize. However, there has been lack of good production because farmers lack quality seeds. Next. My objective was to determine factors affecting access, adoption, and use of improved seed. I was also assessing the efficiency or success of the community-based seed production. And my third objective was to develop an appropriate seed supply system that can produce quality sorghum seeds for people in CIM. Those were my research questions. They were based on the research objectives. Next. Okay, the objective one was a survey conducted in Siaya and specifically in Kagonya, Murumba, Matayos, and Ligala and Sega, because those are the places that we have seed farmers. And a total of 60 farmers were interviewed, and the data collected were agronomic harvest and post-harvest practices. And all this data were the questionnaire was structured so that it can be analyzed using SPSS software. Next. Results. From the results, we can see that most farmers were aged between 41 to 60, and most of them had only primary education. Next. And most of them had no knowledge on sorghum pests. Next. 51% of the farmers, well, 51.6% of the farmers use their own seeds, and others buy from open air markets, and some are issued with seeds from research institutions such as University of Eldoret. So there are no farmers who are getting seeds from certified merchants. And the drying methods they were using after harvesting these seeds were they're using the sun. The farmers dress their seed before storage, and most of them used wood ash for storage, and they store the seeds in room temperature. Next. Most of the farmers reported fertility issues on their farms, and 98.33% had no access to sorghum seed merchants, and they had to travel for more than 4 kilometers for a seed merchant, for those who say that had access. Okay, my objective 2 was to assess the efficiency of successful farmer group and based on sorghum seed production. And the site was Siaya. The varieties were T30B, N4, C26, and E1. Use RCBD, and the treatments were replicated 3 times. Data was collected on N. Sorghum was harvested at N time when the farmers are harvesting, at N plus 7 days, after harvesting at N minus 7 days. That was early. N was used because the varieties matured at different times. So N minus 7 had 30 to 40% moisture content, N had 20 to 29% moisture content, and N plus 7 days had 10 to 19 moisture content. Data was subjected to ANOVA, and objective 2 results. We can see that harvesting at 20 to 29% produced the highest seed vigor. And down there is the moisture content at harvest. We can see that 10 to 19 produces low vigor, and 30 to 40 produced also low vigor. Vigor is an important aspect of seed quality because it determines how the seeds are going to perform in the field. So when we look at the interaction effect between percent germination moisture content at harvest and drying method, we can see that 20 to 29% had the highest germination percent, and drying under the shade had the highest percent germination. Next. Percent germination is different by the way. We can see that shade drying performed well than sun drying. Reason could be that shade drying gave the seeds more time for physiological maturity than sun drying. Sun may have interfered with some metabolic processes in the seed. Next. This is a box plot for moisture content at harvest. We can see that 20 to 29% moisture content had the highest, and the picture on the right shows how the seeds were, some of the seeds were diseased, like from the one below there, harvest at 10 to 19%. When you dry them under the sun and harvest at 10 to 19%, it shows that they had more, they were more diseased than the shade drying. Same is seen when they were harvested at 30 to 40%. Next. So we can, in objective 3, I was testing the demo plots versus the seed farmers plots, and I found out that the demo plots had the highest seed weight than the farmers plot. However, there was no significant difference. So we can conclude that these seed farmers can be used for producing sorghum quality seeds. Next. I concluded that farmers lack access to quality seeds in Siaya, and most of the farmers are uneducated, as most of them were had reached primary level. Shade drying is better than sun drying, as you have seen above, and harvesting at higher and lower moisture content affects seed germination. And we can conclude that CBOs, community-based seed farmers, can be used as a seed source when provided with adequate training. The study recommends that CBOs to be used as seed producers. Farmers need more training on harvest and post-harvest, such as the drying methods, and more studies should be done on the appropriate time of the day to dry their sorghum seeds. Okay, thanks to McKnight funded sorghum project for financial support and School of Agriculture technicians, as well as my supervisors. Next. Those are my references. And the last picture shows when you harvest at early, the consequences of harvesting sorghum early at higher percent moisture. Thank you.
All right, thank you very much, Brian. Sorry for the hitch that was there, but you finished your presentation. I don't know whether anybody could have a question for you. Questions? Well, because we are running out of time, I think we'll thank you, Brian.
If I may.
Yes.
Okay, yeah, Brian, thanks for your presentation. I saw in one of your slides, you harvested with a moisture content of 20 between, gave you the highest seed vigor. What could be the reason? Because I was thinking the moisture content is too high, but your vigor was quite impressive. What could have accounted for that? Could you help? Thank you.
Okay, Brian, you have muted. Can you unmute?
Hello?
Yes, answer the question.
Okay, when you harvest at higher moisture content, the seeds will be diseased. The fungi will interfere with germination. So the seeds, even in storage, the seeds will be diseased, and the seeds will be infected with fungi. And when you harvest at lower moisture, at lower moisture content, this meant that the seeds were left in the field for so long. So they could have been extremely dry, this could have interfered with internal processes such as the germination hormones and things like that. But when you harvest between like for sorghum, it was between 20 to 29. This was found to be the best moisture content for germination and vigor. I don't know whether I've answered your question.
Okay. It's okay.
I think here the issue is when is the physiological maturity? I think Brian needs to be clear on that, because when you harvest between 19 and 30% moisture content, maybe you are harvesting at the right physiological maturity, but then you need to dry the seeds in a better way. That's why drying at such moisture content, when you dry under shade, it takes long, but then you have the right quality of seed. I think Brian needs to be clear there. Thank you very much. I think we should give Anne an opportunity to present.
Hello,
Thank you,
Prof.
Brian.
Thank you, Prof.
Yes. Yes, Anne.
Prof, you can hear me?
Yes, now we can hear you.
Okay.
Okay, there you are, Anne.
Good afternoon. My name is Anne
Afternoon.
Beki. I'm going to present on effect of avascular mycorrhiza fungi, application on phosphorus and copper, availability for improved wheat production in ferrosols. Next. Introduction. Wheat is the second most important cereal grain after maize, mostly grown by large-scale farmers for commercial purposes and for food. Total yield has been on a decline due to low and declining soil fertility. The low soil fertility decreases important nutrients like P and copper, thus making them deficient in the soil. P and copper uptake has to be increased to improve the availability in soil and thus increase its use efficiency and increase wheat grain yield. Next. This is a graph showing trend of wheat grain production from 2007 to 2017. Next. Statement of the problem and justification. There has been a decline of wheat yield during the period whereby the wheat yield grain has been 2.1 tons per hectare against the potential of 6.5 tons per hectare. This is due to the low and declining soil fertility that has caused P and copper deficient. On the right, you can see our wheat plant with P deficient, whereby it has purple coloration, thus leading to stunted plant growth and also few shoots. You can also see a plant with copper deficient, whereby the tips of the plant start dying and thus reducing the tillering and head emergency. This is due to the fixation of the important nutrients due to the soil acidity, where P becomes unavailable to the plants, and also due to the crop harvest, whereby the nutrients are being mined by the plants and it's not replenished when the farmers harvest the crops. Therefore, we are going to use avascular mycorrhiza fungi, whereby this is a fungi that secretes organic acids that dissolves tightly bound minerals like phosphorus, copper, and zinc. The fungi also have hyphae that leads to a network with the plant roots that's to enhance the access of roots to the light soil surfaces. The hyphae are also small and thin that we can be able to penetrate into small spaces between the soil particles. On the right side, you can see a photo where we have a plant root with the hyphae. You can see that the hyphae are very small and they can be able to penetrate into the soil particles. Avascular mycorrhiza fungi improves the plant nutrition by increasing the availability of various nutrients in the soil. That improves nutrient uptake by solubilizing and mobilization and transfer of nutrients. Therefore, the nutrients become available to the soil. Therefore, this will increase soil phosphorus and copper, leading to increase in wheat tillers and head emergency, thus leading to good quality of the grains and thus increasing the wheat grain yield. Next. My broad objective is to evaluate the potential of avascular mycorrhiza in increasing phosphorus and copper use efficiency or wheat grown in ferrosols of Chepkoilel in Uasin Gishu County. The specific objectives are to establish the effect of avascular mycorrhiza fungi on available P and copper in ferrosols of Uasin Gishu County and to investigate the effect of avascular mycorrhiza fungi on wheat growth and grain yield on ferrosols of Uasin Gishu County. Next. I hypothesized that application of avascular mycorrhiza fungi will enhance phosphorus and copper availability for wheat production in ferrosols of Uasin Gishu County, and avascular mycorrhiza fungi will increase wheat growth and grain yield in ferrosols of Uasin Gishu County. Next, materials and methods. The study was carried in Uasin Gishu County, University of Eldoret Farm, with a latitude of 0 degrees 57 minutes north and longitude 35 degrees 30 minutes east, with a rainfall range of 900 to 1,200 millimeters, with a temperature range of high temperature 26 to 17 degrees centigrades, with a soil type of rhodic ferrosols. The experiment was it was a field experiment laid in randomized complete block design factorial arrangement of 3 by 3 by 2, whereby I had 3 rates of P, that is 0, 8.8, and 17.6 P kilograms per hectare, with TSP being the source of P, and 3 rates of copper, that is 0, 5, and 10 kilograms copper per hectare, with copper sulfate being the source of copper, and 2 rates of avascular mycorrhiza fungi, that's being 0 and 60 liters per hectare, with Rhizotech being the, this is the treatment structure, whereby I had control mycorrhiza P at 8.8 kilograms per hectare, 17.6 kilograms per hectare, copper at 5 kilograms copper per hectare, 10 kilograms copper per hectare, and whereby we combined mycorrhiza and phosphorus mycorrhiza with 8.8 kilograms per hectare, mycorrhiza with 17 kilograms per hectare, and mycorrhiza plus 5 kilograms copper per hectare, mycorrhiza with 10 kilograms per hectare and 8.8 kilograms per hectare and 5 kilograms copper per hectare, 8.8 kilograms per hectare and 10 kilograms copper per hectare, 17.6 kilograms per hectare and 5 kilograms copper per hectare, 17.6 kilograms per hectare and 10 kilograms copper per hectare, and whereby we had the 3 combined mycorrhiza with 8.8 kilograms per hectare, 5 kilograms copper per hectare, and mycorrhiza and 8.8 kilograms per hectare, 10 kilograms copper per hectare, mycorrhiza with 17.6 kilograms per hectare and 5 kilograms copper per hectare, mycorrhiza with 17.6 kilograms per hectare and 10 kilograms copper per hectare. The plot size was 2 meters by 2 meters with a spacing of 25 between the rows. The wheat variety that was used was a certified variety from Kenya Seed Company, that is Njoro 2, that is not to be high yielding and does well in fairly acidic soils, and it's also rust resistant. Data collected. We had soil collected that we analyzed for pH, total nitrogen available P, and copper according to Kalebo et al. 2002, wheat grain yield, and also we did statistical analysis. That is my statistical model, whereby we had an effect of phosphorus effect, copper rate effect, avascular mycorrhiza fungi effect, and there was an interaction between phosphorus and copper, and an interaction between phosphorus and avascular mycorrhiza fungi, interaction of copper and avascular mycorrhiza fungi, and the 3 combined. That's the interaction of phosphorus, copper, and avascular mycorrhiza fungi. Next, results. This is a figure showing effect of avascular mycorrhiza on available phosphorus in soils. From this, I'm able to see that the treatment differed significantly in their effect on the soil available P in both the season, that is 2018 and 2019, with the application of avascular mycorrhiza fungi and P at the rate of 17.6 kilograms P per hectare, having the highest amount of soil available P in both seasons. And also, application of avascular mycorrhiza fungi and P at the rate of 17.6 kilograms increased the amount of soil available P compared to avascular mycorrhiza fungi and P at the half rate of 8.8 kilograms per hectare. We can also be able to see that in season 1, that is 2018 and 2019, in 2019, there was a lower residual available P. This could be due to high nutrient uptake by the plants, as it was favored by the higher rainfall that the rainfall was higher in 2019 than in 2018. This can be correlated to the results of grain yield, whereby in 2019, we had a higher grain yield than in 2018. Next. This is a figure showing results on effect of a vascular mycorrhiza on available copper in soils. Here we can be able to see that in the first season, that is 2018, the available copper phosphorus in all the treatments were lower than in the second season, with a vascular mycorrhiza fungi and 10 kilograms copper per hectare having the highest amount of soil available copper. Next. This is a table showing effect of a vascular mycorrhiza fungi, phosphorus, and copper on wheat grain, that is in tons per hectare, during 2018 and 2019 long rain season. Here we can be able to see that combination of, we can be able to see that all the treatments that were combined with a vascular mycorrhiza performed better than the treatments that were not combined with a vascular mycorrhiza fungi. In the first season, we can be able to see that a vascular mycorrhiza fungi combined with 8.8 kilograms per hectare had the highest grain yield, that is in both seasons. In season 1, 2018, we had the highest being 2.8, and in 2019, we had the highest being 4.7 tons per hectare. This being both from the treatment of a vascular mycorrhiza fungi combined with 8.8 kilograms per hectare, that being half the recommended rate. We can also be able to see that there was a 100% yield increase over the control in both seasons. And in season 1, application of AMF led to at least 52% yield above the control. And in season 2, that is 2019, the application of vascular mycorrhiza fungi gave an increase of 97% increase above the control, and use of a vascular mycorrhiza alone had 63% yield above the control. From season 1 and season 2, that is 2018 and 2019, in 2019, the yields were higher. This can be translated to more nutrients removed per unit area, as we have seen from the figure whereby we had low amount of available phosphorus in the soil. Next. Conclusion and recommendation. A vascular mycorrhiza fungi solubilizes P and copper as indicated by higher contents of residual available P whenever a vascular mycorrhiza fungi was applied, thus improving soil P and copper content. Also, the combination of a vascular mycorrhiza fungi and 8.8 kilograms P per hectare had the highest wheat grain yield in both seasons. Recommendation, we can recommend that use of a vascular mycorrhiza fungi with a low rate of P performs better, and that is what should be used by farmers, as high amount of phosphorus reduces the efficacy of AMF. Expected output, at least a publication in a referred journal. And I also would like to acknowledge University of Eldoret, my supervisors and my colleagues. Thank you.
Thank you very much, Anne. Sorry for the hiccup that we have had, but your presentation has been clear and fine. Any questions from the audience? And there are two figures that you have given on P and copper.
Yes.
Able scroll back to those two figures, P and copper, and no results. The next, hapo hizi mbili. For copper, you said that this is available copper in the soil.
Yes.
And you are saying that there was more copper in 2019 than 2018, isn't it?
Not really.
Or rather, that is what your figures are showing, isn't it?
More or less, but mostly on the...
More. According to your figure, the figure that we have there is indicating that there is more in 2019 than in 2018.
2018, yes.
You are not very clear why that is the case, because for P, it is the reverse, isn't it?
Yes.
So what is the explanation for these two results?
For copper, mostly copper applied in soil. According to the research done, once you apply copper in the first year, that can run for 5 years. So most probably, when we added copper in the second season, it was excess because that was just after 2 years.
You should not say probably, Anne, because this is a soil science research. I assume that you did soil analysis before and
Yes,
then after. So you should have a very good explanation as to why this is happening. Do not say probably, because if you had your data correctly, you should be able to have a good reason for that. And then if you go to the previous slide where you have the P, it is indicating that it was is it less? Yes.
The second season.
In the second season.
Yes.
Again, the explanation is not very clear. So you should be able to explain that well, right?
Yeah. I said for phosphorus, there was low residual available P, that is in 2019. This was due to higher nutrient uptake by the plants, as it was favored by the higher rainfalls in 2019 compared to 2018, where the rainfall was less than in 2019. And I said this
if you say there was higher uptake of phosphorus in the second year, then that should also reflect in the plants, isn't it?
Yes.
I hope that reflects, because I'm not sure that that shows. You did not have the results. You did not have the results of plant tissue analysis, etc., etc.
Okay, I have it, but in a different objective.
Okay, so you get my concern, eh?
Yes, I can get, but from this you can even able to see it in the table whereby we have wheat grain yield, whereby in the second season we had higher wheat grain yield than in the first season.
Okay, the yield can explain that, or you should have analyzed. You should have done chemical analysis of the plants.
I'm saying in another objective I have where I have done nutrient composition.
Okay.
Yeah.
All right. So, any other question from anybody else? If there isn't, do we have any question for our 6 presenters? Anyone who has remembered something they want to inquire about? Anyone to ask Julius? Anyone to ask Gerald? Anyone to ask Mr. Bote? And
Prof,
Mami?
maybe I can
Yes,
interject.
Faith, yes.
Yeah, I think I have a question for the first presenter.
First presenter, Julius?
Yes, I think he was talking about fungi.
Yes, the use of fungi to control insects, yes.
Yes, I only had a question in terms of what are the side effects of using these fungi? And the second question was
Side effects
what are,
to who? To the plants or to human beings? Or
yeah, to whoever.
okay, okay,
Yeah.
okay, yes.
Is there any side effects? Because he was saying we are replacing the chemical with the fungi because the
Yes.
chemical has side effects. How about the fungi? Do they have any side effects? And also a second
Beautiful.
question was, are there any challenges in using these fungi?
Yes,
you.
Julius, if you are still there, are there side effects? Just like we have biological control, the insects go out of control and they become pests themselves. How about the fungi? Julius? Julius, are you there? No, we will penalize him next time. I think he's not there. I wish I knew the answer, Faith, I would answer you, but I will inquire and pass over the answer to you later.
Okay, Prof.
Okay, anybody else with a question to Mami or to Brian or to Anne? If there isn't, I want to thank you all very much. We have had very good papers presented in this session. I am sure if we can have some of this uploaded somewhere, most of us will benefit. So I want to thank all the presenters, all the way from Ghana, Senegal, Uganda, and our locals here, and Julius for coming all the way from Chiang Mai University to present here. This is wonderful. Thank you very much, all of you. If you could hear me clapping, I will clap for all of you. Thank you very much.
Thank you. Thank you very much.
I think, yes, we have come to the end of this session, and be blessed. Thank you.
Thank you very much.
Bye-bye.
Bye.
Thank you.
Bye-bye. Thanks, too.
Bye. Bye, Mommy.
Bye, doctor.
The University of Eldoret is a modern university situated approximately 9 kilometers northeast of Eldoret town in Uasin Gishu County, Kenya. Founded in 1946 and converted to a teachers training college in 1984, in 1990 it became Chepkoilel campus of Moi University, offering natural, basic and applied science programs. In August 2010, the campus was upgraded to Chepkoilel University College, a constituent college of Moi University. Upon the award of charter by the President on 11th February 2013, the University College was renamed University of Eldoret. Our vision is a premier university that nurtures global leaders and innovators. Our mission is to provide quality education, training, research and consultancy in science, agriculture and technology to meet the needs and aspirations of a dynamic society. The University of Eldoret is ISO 9001:2015 Quality Management System certified. The university is led by the Chancellor Ambassador Professor Judith Mbula Bahemuka, who is the titular head of the university, the council comprising of the chairperson and members, and the University Management Board led by the Vice Chancellor Professor Teresa Akenga. The other members comprise of Deputy Vice Chancellor Academic and Student Affairs Professor Ruth Otunga, Deputy Vice Chancellor Administration and Finance Professor Wilson Ngetich, Deputy Vice Chancellor Planning, Research and Extension Professor Philip Raburu, the Registrar Academics Professor Paul Taruz, Acting Registrar Administration CPA Francis Ikapel, Registrar Planning Dr. Benjamin Mwasi, Acting Finance Officer CPA Ereu Totona, and Senior Legal Officer Mr. Stephen Anditi. University of Eldoret is science, agriculture and technology based and has grown steadily from the initial schools of forestry and science to the current 9 schools with 34 academic departments. The schools are led by a team of deans who also provide academic leadership and management of the faculty. They include Dr. Oliver Kiplagat, Acting Dean, School of Agriculture and Biotechnology, Professor Robert Otuya, Dean, School of Business and Management Sciences, Dr. Paul Odwori, Dean, School of Economics, Professor Kisilu Kitainge, Dean, School of Education, Dr. Julius Kolongei, Dean, School of Engineering, Professor Gelas Simiyu, Dean, School of Environmental Studies, Dr. Jacob Onyango, Acting Dean, School of Human Resource Development, Professor Boaz Kaunda Arara, Dean, School of Natural Resource Management, Professor Samuel Luta, Dean, School of Science, and the Dean of Students, Dr. Lelei Kiboi.
This is one of the big schools in the University of Eldoret with several programs covering all the fields in agriculture, both at undergraduate level and postgraduate level. Also, we have some diploma programs. We strive to be a center of excellence in agriculture, as our vision is to be a center of excellence locally and globally in agricultural sciences, technology development, and transfer. We have a total of 6 departments within the school, which are under the dean. So we have a wide range of courses, and our students are not limited to work in agriculture, although most of them work in agriculture, but we have other options which are not traditionally agricultural enterprises, but nowadays employ our students.
Yes, good afternoon, everybody. My name is Samuel Awia, and I'm here to present a paper on emerging role of women in household food access in Nyamira North County, Kenya. In Africa, the role of providing for the family is in the hands of men. The culture has actually dedicated that men, they're the ones who are supposed to provide food for the family. And that the provision of food for the family by men actually is a source of social status of men in society. And it is the one that helps men to actually have identity formation. And in this case, men can provide food either by engaging in farming in the household land, or they can actually buy food in some situations if maybe we have food shortage at home. Now, with some of the changes that have occurred in the land system, whereby nowadays in most parts of the country or most parts of Africa, we have the issues of land subdivision, which has led to tremendous decline in household land sizes. And the reduction of land sizes has resulted to reduced food production, and at the end, that has actually heightened hunger in some households. Now, with this particular reduction of food production in the farms because of land subdivision, there's need that now women can join their men in providing for their family, because it is difficult in such situations for only a man to provide for the family, even if the culture provides for that. So therefore, this particular study sought to examine the emerging role of women in household food access and how the emerging role has influenced social lives social life in households in Nyamira North Sub-County. Now, the literature, available literature on household food provision is actually conflicting on who between men and women should provide for the family. A study by Nyawanchi 2016 and Isongoma 1998 and FAO 2011 argues that it is the role, it is the responsibility of men to provide for their family because they are the ones who have resources, because they are the ones who are decision makers, and they're the ones who own land in African context. However, studies like now by Nbongi, Onua, and Hamilton 17, they have argued that women have a role or play a bigger role in household food access, especially in the Western African countries. So therefore, with the diminishing land acreages and the resultant food reduction in the household, now it is not clear whether there's a change in gender roles with regard to household food access. Methodology. This particular study was a qualitative study which was undertaken, which was conducted between July 2021 and October 2021. The study targeted the households in Nyamira North Sub-County, and these particular households, the study focused on the household heads. And the major data collection methods were focus group discussions and in-depth interviews, whereby the flagellers, the chiefs, regional extension officers, and the gender experts from the Department of Gender within the sub-county were involved in such interview. So the data analysis, actually, this particular data that was obtained was coded and analyzed using science and the kind of themes that emerged from the data, but which related to the focus of the study, the emerging role of women in food access in Nyamira North Sub-County. So findings. Now, for us to understand the contribution of the emerging roles of women in household food provision, it was prudent for the study to actually have the background, the historical background on household food provision in the study area. And the study began by looking at the gender dimension in household food provision from the historical perspective. And the finding was that in Nyamira North Sub-County, historically, it has been the role of men to provide food for their families. The culture of the individuals in the study area, which is the culture of the Bagusi community, provided that men, they were the ones responsible.
Recording in progress.
Providing food for their household. And now during the study, but this fund actually noted that it is the household head to ensure that his family has food and other necessities. So a man who don't provide for his family was not respected as a man. That's according to a focus group participant who was 72 years old. And this particular finding concurred with Mbevi and Izogobara 2015, who also argues that the responsibility of providing for the family rests in the domain of men. Now, to the first objective, the matching role of women in household food access. Yeah. Now, this particular study established that there are three emerging roles of women in household food access in the study area. The first emerging role was that women are nowadays involved, or they are nowadays engaged in land use decision-making in the household. That women are nowadays increasingly involved in making how land is being utilized to provide food for the family. Yeah, and this is actually contrary to the culture of the people. But now there were some reasons as to why women are nowadays engaged in household decision, land use decision making. The first one was that some men have moved from the rural areas to the urban areas to look for employment. So therefore, the women, they are the only ones left at home, and they are supposed to manage the land to plant and produce food for the rest of the members, as now they supplement what their husbands are sending. And also the other reason is some men are deeply entrenched in alcoholism. So therefore, they do not have any concern with household food provision. So therefore, the women takes the responsibility to ensure that the household members have food on the table. The other emerging role was improvement in food production. And this one that some women are nowadays engaged in food production. They can either join their husbands in producing that, or they can actually do that one by themselves as their husbands engage in other duties. And this one concurs with the study by Nyamwanchi 2016, who used that women are very critical in household food production and even management within the households. The last emerging role was that engagement in manual work to generate income for household food provision. So the study argues that some women are engaging in doing some manual activities so that they can be paid to buy food for the family. The study by participants argue that these particular women, they can get this particular work in construction sites, in tea estates. Like now, the major cases that they emerged in this particular study was that these women are going to the neighboring Kerito tea estate, when a tea estate is so thick, and other private farms like Nara Berries farm near Sotik. So that is was the matching where most women in this particular area go to do their work. And these particular women, they are actually providing for the family when they get money from this particular work. And this was confirmed during the in-depth interviews that were conducted. Now, how have the emerging role, how has the emerging roles influenced social life in households in the study area? The study establishes that the engagement or improvement of women in household food access has contributed to breakdown of some of the families. And this is because women who sometimes who are providing for the family make some of their husbands to be reluctant, especially the one entrenched in alcoholism. And once they actually come home in the evening and they don't get food, they actually engage in fights. And at the end, some of these particular women move away. They move away, they walk away from their family, from their marriage, so therefore leading to the breakdown of the families. And this was confirmed during a focus group discussion where FGD participants argued that some men have become a burden to society. They are only engaged in alcoholism, and the task of providing for their families has been left to women. If they fail to get food after a day-long drinking, they resort to fights, and this has forced some women to leave their marriages. And this is according to an FGD participant who was a female for the 3 years. And the other reason as to why we have the breakdown of families that some women have been compelled by the situations at home to go and look for employment away from home, especially in the tea estates. And while there, they actually sometimes, some of them, they have ended up remarrying or engaging in other relationships that has contributed to the breakdown of their families. So the breakdown of the families is one of the how the emerging role of women in the household food provision has contributed to household social life. The second one, we have diminished respect for husbands, because traditionally or culturally, men are the ones who are supposed to provide for the family. And if now it has changed to that it is the women who are providing for this particular male, the respect that men commanded when they were providing food it is declining because of that particular situation. And as it emerged in the focus group discussion, is that a participant is saying that a man who fails to provide for his family commands limited respect from her wife. How can a woman respect a husband who does not participate in any way in household food provision? So because of non-provisional nature of this particular men, that is how now the respect has gone down from their wives, and this is actually contrary to what was what was there sometimes back, and according to the culture of the people. And lastly, we have witnessed a change in household power relations. Yeah, that the engagement of women in household food provision is solely fostering women's role in household decision making. And these particular decisions may be in terms of making decisions on how children can go to school, how to get food, land use management, among others. And in some situations, some women are making the decisions on how to use land and manage food in the household. And during the FGD discussion, one of the participants argued that the emerging issue of women participation in household food provision is slowly changing power dynamics in households. Power is slowly shifting to women who are nowadays making some major decisions in households. Yeah, this is adversely eroding patriarchal power men have enjoyed over time. So because of the women taking a center stage in providing for their families, now the power that men have enjoyed over time is slowly eroding. That's why you get women making decisions on land use management in the households. That's how you get women making decisions on how to provide food for the family, among other ways. So that is how the emerging roles of women food provision in the household has resulted in a change in social life in households. Conclusion. The study concludes that women have taken an active role in household food provision. So they have assumed the role of land use decision making, involvement in food production in the farms, and engagement in manual activities to generate income so as to provide food for the family. However, such engagement of women in household food provision has resulted in immense social change in society, whereby in households we have it has affected household integration in the family, respect for household heads, and it has led to changes in household power relations. So that some of those are the findings of this particular study. Thank you.
Thank you very much for your very interesting study. I now open the field, or I now ask if you have any question for our presenter. It is now your time. You can show by raising your hand, then you'll be given opportunity to ask your question. Anyone with a question? I think that let me ask one or two which I have.
Yes.
Samuel, my first question is why did you choose qualitative analysis as opposed to quantitative?
Thank you very much. The reason as to why I choose qualitative is that the issue of culture, or the issue of the gender roles, they are naturally qualitative, because I wanted to understand the insight, the in-depth information about how this particular the changing role has influenced the social life, and how the women are nowadays engaged in household.
Hello, we can't hear you.
I'm saying that the reason as to why I use qualitative is that the things, or the study objectives were actually, they could be brought out clearly using the qualitative study, because the qualitative study would actually permit or give the study a leeway to have in-depth interviews or in-depth understanding to the perceptions and opinions of the people regarding how the emerging roles of women in household food provision. And this one would not be provided by the quantitative, because the issues are in terms of attitudes and opinions of the people. So therefore, qualitative was the best suited for this particular study.
Thank you, Samuel. Anyone else with a question? Okay, then let's move on to the next presenter, Omuse.
Thank you.
You don't have your slides. Are you able to share them? You can be given sharing rights. Omse? Or would you prefer to send it to us so that we send to you? Okay. Okay, Ayua, we need you to stop sharing, please. Ayua, Samuel, stop sharing your screen. Thank you. Thank you. So you can now share your screen. Okay, as Omuse tries to share his screen, Ayua, don't go away. There might be someone with a question at the end of the session. So just stick around so that you may be able to respond to your questions. Thank you.
Thank you.
Okay, Omuse, we are waiting for you, but if you are unable to do so, you can send your slides to us. so that we share for you. Almost there. Yes, disappeared. See, we have Philomena. Can we move to the next as we wait for Muse to sort out those issues? Philomena, what are you presenting? Hello, Philomena.
Unmute. Hello, Chair.
Hello, we want you to present as Omuse.
Hello, I am Philomena. I hear you calling Philomena.
Yes.
I presented mine at 11.
Oh,
at 11.
okay. Okay. I see
Yeah.
our ICT are looking at yours right now. Okay, let me then see what next. Ameshindwa kushare vitu zake, what he wants to do with that. Ni mteja huyu, hata tumemcall na mteja.
including Margaret Ngigi and Gideon Obare. This presentation is on determinant of food security through self-sufficient production among maize farmers in Western Kenya. Next. Next slide, please. Now, since independence, and in fact, since when we had the Siderton Plan of 1952, the country has been looking at the way of fighting hunger. And in the recent years, we look at it more on the issue of food security. We have had a number of plans, starting with the one I've just talked about, the Siderton Plan. Then we had the session paper number 10. We have, after every 10 years, we have had other plans until we go to 2008 when we developed the Vision 2030. And later we have had, within the agricultural sector, we have had a number of plans which are looking at how the smallholding farming system, which we have adopted, can be enhanced for food production, first to meet food sufficient, and later provide income which will sustain the smallholders to be rich. Next, please. One of those crops which has been quite adopted is maize, which is grown from all the agroecological zones, starting from coast. There is a variety come to the lower eastern. We have the Katumani, going as high as the higher places, the Timbolo and Mount Elgon. We have had maize variety for those places, and despite all that effort, we realize that food security has been said to be a problem. And through that, a number of agencies have developed research programs to come and intervene. So in this perspective, we may say despite food security being a key milestone associated with sustainable development goals, the sustainable development goals which were developed by the United Nations, ensuring food security, especially in developing countries, has remained a major or an insurmountable challenge. Next. So with this in mind, we set out to look at our small-scale farmers, and we went out to Western Kenya. There is one unique thing with Western Kenya. The first thing is that a large number of we have a large number of our farmers dealing with maize beans intercrop. Then Western Kenya initially had very rich soils, including the ferrosos, and which were volcanic in nature. The place received sufficient rainfall, and definitely we expect that these areas should lead in ensuring that farmers are food secure. However, research queries have remained on if that place, the Western Kenya, farmers are able to meet their food demand. And the other question which we were looking to address is are smallholder maize producers meeting their food availability through self-sufficient production. As I said earlier, the whole aspect is to look at, they first feed themselves, and what remains as surplus is sold for profit. So the food security aspect will also give you on the issue of income. Next. So the general objective of this research is to evaluate smallholder maize producers' achievement of the 4 key aspects of food security, including availability, access, utilization, and stability. The key issue first is availability, because you can't utilize what you don't have. So basically, we went out and we set to look at that aspect of availability. So the specific objective were to determine the socioeconomic and agricultural factors affecting achievement of food sufficiency through own production in Western Kenya. And the other objective was to characterize smallholder maize producers' food availability through self-sufficient in Western Kenya. Next. Next page, please. The method, we surveyed 400 small-scale farmers In the quarter 1 of 2018, that is between January and March in 2018, in some heterogeneous cluster sub-counties in Western Kenya. If you come to the next slide, I'll show you how we settled on the heterogeneity. Then a multi-sampling research including purposive and random sampling were adopted, where we settled on Mount Elgon, Sabatia, Bumula, and Bungoma North sub-counties. A questionnaire was used in data collection. Next, please. Now, I talked about heterogeneity in agricultural production and also the socioeconomic characteristics of the farmers. If you look at that table, we have characteristics from agroecological zones, where the sampled sub-countries were in different agroecological zones. We talk about altitude, annual temperatures, mean annual rainfall, cereal growing days, which was an important aspect to determine the number of labor days. Then the soil characteristics and the population density. The population density definitely gave us the acreage per person or per farmer. Next, please. Now, those are the sub-county we settled on. And out of the entire Western Kenya, we had 3 sub-counties in Bungoma, that is Bungoma North, Mount Elgon, and Bumura. And the other sub-county was in Sabatia in Vihiga sub-county. Next. Descriptive research output are presented in terms of tables and graphs. And then we also undertook an ordinary least square OLS linear model. Outcome was used analytically to relate food sufficiency with both socioeconomic and agricultural characteristics. Next. Results and discussion. And I'll start first with the characteristics of characterizing household socioeconomic characteristics against food sufficiency. And now what you look at in that table is that we have the socioeconomic characteristics, and the third thing was the level of staple food sufficient overall in Mount Elgon, Bungoma North, Bumula, and Sabatia. What you realize there is that in Sabatia, level of food sufficiency is quite low at 56%. Then we had years of household head years in total. That was telling us how old they are. You realize farmers in Sabatia, they are relatively older than the other sub-counties. We had experience in farming. Still, Sabatia had some old farmers. Household size, we had Mount Elgon having the largest population. Then we had adult equivalent. This is a measure of how much food they needed in total. And this is an indices which is used to give a person food requirement by age and by sex. And what we did is to do the summation per household. Then we have the issue of land owned and per capita expenses within a week. And what you realize is that there were so low, the amount spent per person per day was quite low. Because as for other document on poverty, somebody is to be below poverty line, you are supposed to spend as low as anybody who spent less than 200 per day is considered poor. Next. These are the agricultural characteristics associated with food sufficient in Western Kenya. We had the amount of food produced. And what we realized is that in 2017, because we did the study in 2018, so we were looking at aspects to do with the 2017, you realize that per household, Sabatia produced the least, about 450 kilograms of maize per year. And also the land allocated to maize was quite low, around 0.2 hectares. And basically this is because of the the high density, which meant that the land available for agricultural production was quite low. Next. There is other information to do with agricultural production, that table. Now, Now, the outcome of this study, if you look at that graph, you realize that whatever is in green is when farmers were claiming they didn't have sufficient food. The food was insufficient, that is the staple, which is maize in this case. Then the red, which look like lead, is basically those who had a lot of concern about food. They could have had food, but they were forced to use it in a very, very economical way to ensure that they have the next for some little for the next day. Then we have sufficient amount, and that graph shows the little green picture of food security in Western Kenya, where you only realize that is only the highest when we had the highest proportion of people believing to be having food sufficient was 18%. That was in September, but from August to December, it was 16%. Then looking at that graph, you realize there is the aspect of when maize is produced. And in this case, maize is produced, they start harvesting in August, heading to December. By the time they are done with the December, the issue of a large number of people reporting both insufficient in food and food availability concern really rose to very high rate, as high as 91%. Remember, these guys are supposed to produce maize. They feed themselves first and then they sell to get income. So definitely the picture of food insecurity is also a replica of economic or financial insecurity. Next. Next. So let's look at maize production, purchase, use, and diversification among households. If you look at that graph, the first one in blue indicates it's just the trend of, maybe you could go up a bit, sorry, just a bit, up. Up a bit, please. Up, not down, please, up. If you look at, there. If you look at that graph, maize own production around go down a bit, sorry, you have covered the upper part, but in real sense you see that, okay, that graph shows the trend in maize production, and what you see is that between August and December, around 80% of the smallholders are using their own food, what they produce. And then after that season, you also see that the use of other staples is very low, the lower graph, which is in purple. Yes, I think that is purple. Yes, it's in purple. Then around the line which is in green, it shows the amount consumed per month. That is in kilogram of maize. And what you realize is that on average, around between September and December, a high amount of maize per unit is used, which is higher than around April and July. So in a nutshell, what you will say is that graph shows the trend in food availability. And as it has been said in other documentation, in this smallholder production system, we always have what is called the maize, the hunger month. And this month could be considered to range from March to June. When you realize like most of the farmers are purchasing maize, they are using little quantity, and a very small proportion are using their own produce as food. Next. And then within it, you realize that farmers are tending to go to other staples, because by the end of the day, if you don't have maize and you have to buy, you also have to consider what is cheap in the market. Now, sufficiency in food production among smallholder farmers in Western Kenya, and that is what that graph shows. The first one, the brew, that is what is indicated is around, they are able to meet their food and they sell the extra. Overall, you realize there's only about 33% who are able to do that. And in Bungoma North is about 60. If you look at the graph in Sabatia, you realize a large number of farmers are only able to meet between 50 to 75% of their food. And this percent is also relatively low because it's about 37. A large number of farmers and are just between, also a significant proportion of farmers are able to produce less than 25% of maize, especially in Sabatia. Next, please. So when we related, we tried to relate the socioeconomic characteristics, socioeconomic and agricultural characteristics to food sufficient. This is what we observed. That next page, please. There, enough. What you realize is that a number of factors were considered in this regression. We have household head gender. That is whether the household head was a female or a male. The age of household head. Then farming experience. We had issues to do with whether they are formally employed or not. And the issue of adult equivalent, and then per capita expenditure, which is a measure of income, expenditure spent. We had credit, and what was significant was that the household monthly use. Those households which were using, if you look at it, it shows the coefficient is negative. So those who are using higher amount of maize per month were the probability of being food sufficient or insufficient was high. And then the other issue which was significant at 1% is the tropical livestock unit. This Daventry was a measure of the amount of, or the total livestock which they had. It uses indices and it includes livestock which is given a certain value, goats, sheep, and chicken. They are given a certain value, and through that, you are able to know what amount, what goes on. However, in that indices, the issue of tropical livestock unit, cattle, because of its size and its weight, it really influences the outcome. The other factor at 5%, the significant socioeconomic factor which was affecting food sufficient was household head level of education. It was positive. So as people become well educated, they were able to meet the food demands through own food production. Next. Next. So still from there, we had the regional aspects. And what you see, the baseline was Mount Elgon. Maybe you go, the baseline was Mount Elgon. And what you realize is that as you head from Mount Elgon, if you move to Bungoma North, the probability of finding farmers food secure through own production was high, but it was also significant as you headed toward Sabatia, where you could realize that farmers were, the probability of a household being food insecure or being unable to meet their food demand was high at significant at 1% significant level, while at 5% significant level, you could realize that as you head to Bungoma North, farmers were becoming food secure through own food production. Let's look at the conclusion. What we say that despite smallholder farmer being involved in subsistence farming, where the aim is agricultural production for food sufficient first and supply produce for selling, we realize a large proportion of farmers, as high as 66%, were unable to meet their food needs. The other thing we realize is that a place like Vihiga, despite its agro characteristics, agroecological and socioeconomic characteristics, is only a tenth of the farmers or smallholders who are able to meet their food demand through own food production. Next. Next, please. Next slide, please. Then we also realized that there was severe, if you look at the monthly perspective, severe and endemic food insecurity prevails in Western Kenya, calling for technological, policy, and information dissemination intervention that will mitigate the observed food challenges. What we went for, the designing of this paper was more from the policy perspective. And when we talk about technological, if farmers in Vihiga have 0.2 hectares of land on maize, and there are many, we definitely knew that there is that farm itself will require better technologies, maybe beyond what has been recommended, the best seed, and the fertilizers, so that you ensure that farmers are able to meet their food demand and also get extra to sell. Then the other aspect is that food availability status was associated with the duration of the year as influenced by agricultural production seasons, where you realize around some month there is enough food, around other month there is no enough food. And this goes also to consider the large number of people who are able to meet their food demand. And that aspect by itself, we expected it to have very significant factors in influencing other on-farm employment opportunities and the welfare of the farmers. Next. Recommendation, we say that farmers should adopt the high-yielding agricultural technologies so that they could boost their staple food production, because there are some which are there, and consequently increase food self-sufficient in all months of the year and income from surplus. We also recommend that the government and other development agents, the governments, we have the national and the county and other development agencies, including research institutions, international organizations, private sector actors, should develop and promote technologies associated with increased food production and storage. What our thinking here is that we still need to develop better technologies for farmers to use. They need to be promoted, and we have to consider the aspect of storage. If you have to ensure a farmer have food, because that's what goes on, they keep a little, whatever they have, from September to June, you have to ensure that they have better storage facilities. So thank you very much. That is the end. Thank you very much.
Okay, thank you, Mr. Maura, for a very insightful look into food security. We now want to open the floor for any questions. You can raise your hand or you can put it in the chat. Welcome, participants. Let's be active. There seems to be else being put in the ICT can remove that before we go to. We need to interrogate Mr. Maura's work. Let me kickstart that aspect. Mr. Maura, you talked about other staples. Maybe you could give us examples of other staples which replace maize during those times when there is scarcity of maize.
Do I need to intercede right away, or I can get a number of questions so that I answer them concurrently, hoping they are there anyway? Chair?
Okay, let me give you all the questions that I have. Then you can answer them.
Okay, fine. Thank you.
Yeah. So the first one was about what are the other staples you are talking about during those months of scarcity. And then another one was to do with your study concentrated mainly on maize, but I know, or I think food security is also to do with a balanced diet. I don't know if you can comment on that, give us another look at that. And then I was also having the question, how did you measure food sufficiency? What tools did you use to do that? Then the third one is about the other factors which are not significant. I was wondering the many factors in your linear regression that were not significant. We explained the significant ones, but it's also an interesting look at why can't gender affect, what could be the reason. Maybe you can pick a few and just try to explain to us so that we understand. Maybe it is because of the area in which you did your research. Lastly, the other one is to do with your recommendations. You have already told us that the land was 0.1. Okay. Sorry.
Yes, 0.2%. Hello?
I'm back.
Yes, Chair. Oh, you are back.
Yes. Now, you said
Recommendations?
the land,
0.2
about the land. Yes, you said that the land is 0 point something. Yeah. I think it is too small for it to produce much.
Yes.
So when you talk about technological aspects and maize, I try to think out of. I'm trying to think which maize variety would improve its productivity in such a small piece of land. So I was wondering, maybe I can comment on that and say that, like saying they can turn to other high value crops which can produce very well in a small piece of land. That's what I was thinking, as not really there could not be a technology which maybe can improve productivity for maize especially. I don't know, you'll comment on that. And the same also with the policy. I was wondering which policy, maybe you can elaborate a bit. And then also the information dissemination, maybe what could that be? But as a last one is maybe a comment. When you talked about livestock being significant in your linear regression, I thought maybe it could also be as a result of them using the livestock to plow the land. I was just imagining. Maybe that could be why there was a significance. Maybe they had better yields because of that. Okay, so many stories. Let me stop there. You can
Okay,
answer.
thank you.
Yeah.
Yes, thank you very much for those questions. I'll start with the first staple. And maybe it will answer in a way, I'll also tackle the third. Okay, we all know we also have our views of what happens in Western Kenya. And we know maize is the staple and is also the main meal. And a large number of studies, which I could not highlight from the word go, have been talking about that. And it got to some times where guys have come and said every family member requires 2 bags of maize per year. There are all those recommendations in literature. I'll not talk about that. However, what you realize is that when there is the insufficient of maize, people go for rice. There is sweet potato. There is also beans become around June. when beans comes out as the first crop, you almost realize that it becomes like it's the staple. Even if you talk to people, we know beans is part of what is added to food, but basically, in this case, they were referring it as staple. So I've discussed those three: rice, sweet potato, and beans. And if you could go to Mount Elgon, the issue of Irish potato was coming in to replace maize. However, when maize was there, Irish potato was sold. Now, you have said I mainly dealt with maize as a measure of food security. Yes, what we wanted to do is that a lot of effort has been done on maize. In fact, you find maize for every place, and farmers try all type of maize in every place. So in Kenya, even when the government is looking at the issue of food security, it's all the issue of food availability, they are considering maize. So basically what we used is maize for production, because it's first is a food crop, next is a cash crop, and also is widely used. It may not be the most balanced way of measuring from the socioeconomic aspect or from a food nutritionist, but basically for us in economics and looking at agricultural production against the crop, without maize would really give us that aspect of food security. And that's why I talked from the word go, I'm talking about food availability. One aspect of looking at the other, there are four other aspects of looking at food security, but we looked at one enterprise, food availability, and this being maize. So how did we measure food sufficiency? What we looked at is out of the maize a farmer produced, or whatever they were using, what proportion was used from their own production. And that's what you realize, that in some times we were saying most of the food was bought, most of the maize was bought, and our measure was just like, are you producing sufficient maize to take you through the year? Moreover, your work is as a farmer to produce maize, so that first you meet the food availability requirement and next you sell. So that's what I looked at. The other aspect is other factors which were not significant. You felt I needed to have highlighted a number of them. Yes, there is the issue like of gender, and what you realize is that the household, just looking at it, it's a negative perspective, it's a negative coefficient, and 0 is for the lady and 1 is for female. 1 is for male-headed, 0 is for female-headed. And what you realize, although it was not significant, is that for the probability of having a female head of family had really influenced the possibility of being food secure. Then the next one, as the age of the farmer, as farmers were becoming older, the food availability, ability to produce enough to meet their family was a bit restricted. That's also happened with the experience in farming. There is one feeling in social science that as farmers farm more, they become more experienced and they become better farmers. However, what you realize is that it means that as they get more years of experience, there is that challenge of being able to produce enough. Maybe it's because of age. Maybe age is catching up of them, they don't have energy, and all those aspects are working against them. Lastly, I look at the issue of formal employment. If you look at it, it's positive. One, as farmers are more formally employed, they are able to, the probability, it's not significant, but the probability of a farmer who is formally employed to have food sufficient is high. This is maybe because from employment they can get more inputs, and from employment they get more money, and they are not, at times farmers sell their maize, so for them they are not able to sell, they can't sell because they have other sources of income. The membership to farmer groups is also a critical one because mostly extension now is being looked at from the farmer group perspective. And what we realize, those farmers who are not members of a group have better probabilities of meeting their food availability. Then there is another aspect you talked about, the livestock unit. Yes, the tropical livestock unit, where you are saying maybe what happens with the tropical livestock unit is that those farmers who are able to get money, who are well up, they can even have more livestock, and through that, because they're able also to feed themselves, they can have extra livestock. It may be an enhanced lifestyle by the virtue that you are well off. Then the recommendation is that the 0.2 hectares of land, yes, we did a general recommendation. First, we were cognizant that the fact that there are some guys like in other sub-counties, except Sabatia, who had bigger farms. And then we were looking at an aspect that if you look at farming from a socioeconomic perspective, farmers get there first to be busy or for employment. And basically you can't go and tell people to stop farming because they are not meeting their return. The best thing is to tell them to adopt better technologies, and when they adopt, at least the little they get is it saves on money, and it enhances food security. If you look at the 10% of the farmers in Sabatia, they are still operating on an average of 0.2 hectares, but they are able to meet their food demand. That's why we went strategically to say first, enhance technology adoption, and we said develop because they can still be developed, and then after that, we were looking at the government to intervene in a number of factors. Because some of these guys with small farm, they are supposed to be exiting agriculture, but it's not an easy thing to just tell them to quit. Basically, normally what happens, if you look at those areas in Sabatia, they have small pieces of land and tea, small pieces of land and maize. They also have the small, the land which is allocated to the house. And we thought this is the most fair way of giving that recommendation, which is practical, and then it will make people participate in the decision on their livelihood. Otherwise, thank you very much.
Thank you, Mr. Maura. I think you've answered very well. I now at least have a good idea of those questions that I had. And I'm sure also the participants have gotten some more insight into your work. We now want to move on to Omuse. I think we have managed to get your presentation. So we are going to share your slide. Okay, you will show us in the chat, so because you will be our last presenter, so prepare. But for now, let's go to Omuse. The floor is yours. You have 15 minutes. Please mute if you are with someone who needs to be mute.
Okay. Thank you, Chair. Can you hear me?
Just a minute. Francis needs to be muted.
Hello.
Hello, we can hear you.
Oh, you can hear me. That's great.
Yes, yes. Let's
okay,
continue.
Thank you very much. My name is Dr. Emoit Omuse, and I'm presenting a paper on prevalence of food insecurity among university students. And of course, looking at Uasin Gishu County in Kenya. And I'm doing this on behalf of Professor Ruth Otunga.
Just a minute.
Yes.
We need, I think, have you muted? Okay, thank you. Now you can begin again, sorry.
Okay, thank you, Chair. I was saying, my name is Dr. Emoit Omuse, and I'm making a presentation on the topic prevalence of food insecurity among university students, and of course, looking at Uasin Gishu, the situation in Uasin Gishu County in Kenya. And I'm doing this on behalf of the other contributors, Professor Ruth Otunga, who was our principal investigator in this project. Then we have Dr. Lelei Kiboi, Dr. Rachel Koros, Dr. Makomere, and Mr. Mwangi. Can we move on? Okay, so we were looking at this topic, like I've already mentioned, against the backdrop of the food insecurity, particularly in our part of the world, that's the third world, and particularly Kenya. But of course, the focus was on what takes place in Uasin Gishu County. So generally, we looked at two institutions in Uasin Gishu County, which is Moi University and University of Eldoret, so that we wanted to understand what students go through in terms of being food secure, which in our assessment is a determinant to academic performance. So we were relating the issue of food vis-a-vis academic performance. So of course, we are aware that the economy in Kenya is not doing very well. Then there's also reduced government capitation in universities, bringing about a high cost of tuition fees and the other costs of living. So these, particularly these two, the economic downturn, then reduced capitation over years, especially the last few years, has resulted into hardships among students, and therefore the net effect of this is challenges to do with food. Of course, globally, you realize that Africa is disadvantaged. When you look at the hunger index, Africa does very badly compared to other continents. In our case in Kenya here, even before we look at institutions, then you realize that almost every year there is a food alert. There is a hunger alert by the Red Cross, and that one trickles down to institutions of learning too. So in our presentation, we have looked at the possible factors. We identified factors that affect food situation among students. Of course, affects everybody, but also it comes down to the students. We look at population density, and of course, against the available food, then there is a problem there. Then we know that there have been climate, the patterns have changed over years, making it very difficult for consistent food production in the country, and that one brings hunger. Then now we have urbanization, which has eaten into farms that hitherto are used for agriculture nowadays have been turned into commercial land. Then insecurity, particularly resulting from terrorism, it affects the way the whole farming enterprise is undertaken. And of course, all these factors, like I've said, affect the society generally, and it comes down to students, because society funds education. It's the society that supports the students in school, and therefore, if the society is affected, then that one comes down to the students. Then, of course, before this, we had just tried to figure out, to understand, when you talk about food insecurity, then what exactly do we have in mind? Then generally we are saying this is a situation where students, because we are looking at the context of the students, so that's why I'll focus on that more, is where students lack nutritionally adequate food that is not really safe or limited. And like I said when I started this presentation, you relate the availability of food, or when somebody is food secure, you relate it to academic performance. So the lack of this, and particularly if the nutritional value to that particular food is wanting, then that one has the potential for affecting a student. So that's how we try to look at that particular definition. Then in our study, we looked at, we employed a mixed method research design. Of course, we incorporated both qualitative and quantitative approach of inquiry. And we utilized the questionnaires. We interviewed particularly deans of students in the 2 institutions. Then we also did some focus group discussion. Then we analyzed documents. Then, of course, data resulting from this was both qualitative and quantitative. We employed random sampling to collect data from information. Then, of course, there were participants that we went direct to them. For example, the dean of students, you just go direct. There are 2 deans of students in the 2 institutions. So we employed purposive sampling in this study. Then, of course, before we look at the findings, we reviewed literature on other works that people have done relating to issues of food insecurity, globally, of course, and of course, locally too. We looked at works from the African continent, especially South Africa, and you look at people who have done this kind of related work in South Africa, then their conclusion is that they attribute almost 40% of academic challenges in South Africa to the challenges brought about by food insecurity. Then globally too, we looked at some studies funded by World Bank. And generally in all these studies, you critique them, then the conclusion is that food insecurity is a serious problem that affects society in general, but of course, that effect finds its way to students in institutions of learning. Recent research, for example, has indicated that food insecurity has been on the increase among university students from both incidentally developed as well as developing countries. And maybe again, I would like to take you to the definition. We've said it's not just a lack of food, but it also indicates the nutritional value of the food available maybe low. So that's how we say that it's a problem in the developed as well as developing countries. But of course, the problem is pronounced in African continent. Looking at our own Kenyan situation, then you can appreciate and understand that. Let's move on to the findings of the research. So we looked at how the magnitude of this problem, prevalence of food insecurity among students. And again, I would just like to remind the listeners that we focused at University of Eldoret and UoE university. Then when you look at this particular component, the findings indicated that 51% of students indicated that they were not secure in terms of food. Then, of course, 47, you would argue that those ones who are insecure versus those who are secure, the percentage is small, but we are looking at the net effect of even the slightest insufficiency on the learners. Then you discover that there is a big problem. And of course, the implication of this is that majority of the learners in these two institutions do not have enough supply of food. Then we also try to find out how many meals these students have per day. Of course, looking at the definition of the number of meals in our part of the world, then we normally have in mind breakfast, then lunch and supper. That's the basic number of meals that we have. Then you look at this, then the percentages there show that 46.2 of the students skipped 1 or 2 meals. Then, of course, the implication of that is that the majority of the students have a single meal a day. And again, relating this to academic performance, then that one must have an effect, because either a student starts the day before without a meal, or ends the day without a meal, or skips a meal in the course of the day. And of course, learning goes on through. Then on whether these students had a choice when it comes to meals, then it follows the same trend, because if you skip 1 or 2 meals, then what that means that you do not have the luxury of choice here. So you take whatever is there, and that one is indicated by the percentage there, 65.8% indicated that they took whatever was available. And therefore, whether it was nutritionally adequate, whether it was enough in terms of amount, that was not important, but whatever was available, students took that. Then a second finding relating to this study, we looked at what are the possible factors contributing to food insecurity. Then the responses are indicated there. 29.4% attributed this to financial instability. What are the sources of finance to students? Of course, we have parents, we have well-wishers, we have the government through High Education Loans Board, we have Constituency Development Fund. Those are some of the funding agencies and, of course, persons to students. So financial instability, for example, when you look at the case of HELB in our main document, the provision of funding from HELB keeps on fluctuating, and sometimes it comes very late. Sometimes some students miss it out in subsequent years. So you see that instability brings about lack of funds, and therefore translating into inability of the students to get food while in campus. Then 53.8% attributed this to family backgrounds. Of course, we know when the families struggle them, it becomes also difficult for them to fund adequately the students, and that one affects the learners. Then there was that percentage, 44%. That one is attributed to individual habits, inability to budget for the available resources. Affected students contributed to inability to get food. Then the rising cost of living at 44% contributed to food insecurity. And we know what has been happening, especially against the backdrop of the pandemic and other factors, how the cost of living has been skyrocketing over the last 2 years. So that one affects students. Yes.
Please kindly summarize your 15 minutes.
Okay, all right. That's exactly what I'm doing. Then, of course, significant among this one was poor family background, which, according to our study, contributed highly to this instability. Then we also looked at the component of effects of food insecurity on academic performance. Just move on. Move on. Yes. We found out that 8.2% of the students deferred their studies because of food, which means deferment is not as a result of food. Then 56% agreed that food insecurity affected academic performance. So generally here, you note that academic performance, attendance to lectures, and of course participation in all academic activities is affected by food insecurity. Let's move on. Move on, please. Then we have other factors contributing to this. For example, behavior among learners. Then we have absenteeism, psychosocial dysfunction, and low scores. All these ones are effects of food insecurity. Let's continue. So what we concluded in this study was that the food situation in universities, among university students, is dire, and it affects performance, academic performance. That was our conclusion. Let's move on to recommendations. Of course, we made recommendations against what we found out. Number one, for example, we have said universities or institutions of higher learning can improve, can enhance their work study so that as many students as possible can come in, and therefore that one can help them to secure food. Then we also said students should be supported so that they don't defer their studies. Then number three, we have said that institutions can liaise with funding agencies, sponsors, so that students can be supported. And that one will bring about academic stability because they'll be able to take care of their food needs. Let's continue. Then related to that, universities can also liaise with charitable organizations so that students benefit on tuition issues. Then we also recommend that career offices in the Dean of Students offices should liaise with potential local employers so that students can be incorporated in their work programs, and that one can really lift their economic status, making them food secure. Then counseling is very important, especially from our study, we say that students are unable to manage their finances, and therefore counseling is very important so that students are helped on financial management so that they can budget on the little resources that they may have. Thank you so much, Chair. That is the end of my presentation.
Thank you so much, Dr. Musa. It's a really important aspect of what we do as universities, for us to know how our students are doing and to be able to have an explanation as to the situations, the class performance we are seeing. So I think it was a very important study to do. I open the floor to any questions anyone may have. Kindly let's participate. Can ask any question. You have the expert here, because he's done his study. I'm sure you can at least, even if it's a little bit out, it will still be good to know. Okay, let me kickstart because I can see our last presenter is here. I just wanted to inquire about the number of students you interviewed. Second thing is, I think I felt in your recommendation, you could maybe have recommended that we go back to the university system we used to have, where food was provided by the government. I think I was under such a system, and I think that really helped a lot of low income families who had brought their children to the university. So maybe I thought that would be a recommendation. After all, we are going with the free health services or universal health services, and all these other things are becoming free. Why not even providing for our university students? So I know that's a really complex, but maybe that would be my input there. So I've asked the number of students, and also I have an inquiry on how unique is the food insecurity experienced by the students. That one I think you have answered. So maybe just how many students and whatever else you want to comment on.
Okay, thank you, Chair. Maybe let me begin with the one you have asked, and of course, made also a contribution that it could be a very challenging issue, recommendation regarding reverting back to the older system, feeding students. And I think we are happy for you. You enjoyed that privilege where you had the luxury of choice. And of course, you could also walk into the mess. Of course, those times were quite friendly to the students. But we must agree that circumstances have really changed. We talked about economic downturn. We talked about other issues, population, like that. Maybe just to talk about the older system vis-a-vis the number of institutions and the students who are getting to university, then you realize at that time the number of students getting to university was manageable. But since the opening up of higher education, then you realize there are so many students coming to university, such that even the funding agency, the HELB, is overwhelmed. Students cannot be given adequate funding. And therefore, that recommendation is good, but of course also looking at the reality of the day. Naturally, we make recommendations that are realistic, and that one may not. Okay, you can make a proposal, you can recommend, but the prevailing circumstances cannot maybe take us back to the good old days that were there. The number of students, we used a sample of random sampling to select 4,400 students. And therefore, our kind of study, generally that one constituted 10%, which is acceptable in our kind of study. I didn't quite get your last comment, Chair.
I just noted that as you were presenting, I was trying to think what is so unique about the food insecurity being experienced by students, but you actually answered it as you went, because you gave the reasons as to why they don't have enough food.
Okay.
Yeah.
Fine.
Okay.
Thank you. Thank you.
Yeah. Thank you for your presentation. I think we'll move on because of time. Unless there's somebody with a question, you can put it in the chat, and I'm sure Daktari will be very willing to answer those questions. We now want to move on to Frederick Agego, and we are talking about efficacy of composting with snail meat powder on protein nutrition quality of sorghum wheat bran using a rat bioassay. It sounds very interesting. I hope the last word rat doesn't mean what I think. So you're welcome, Frederick.
Thank you so much. I hope you can hear me.
Yes, we can.
Thank you. My study is on the efficacy of compositing with snail meat powder on protein nutritional quality of sorghum wheat bands using a rat bioassay. This work has been done in collaboration with other authors, Professor Arnold Onyango, Dr. Charlotte Serem, and Dr. Judith Okoth. Let's move to slide 1, the introduction. Okay. Yeah, there. So in sub-Saharan Africa, there are more than 220 million persons who are undernourished. This is because of poverty and overdependence on those diets which are cereal-based. And these diets are poor in nutritional in protein quality that normally exposes children to protein energy malnutrition. So in developing country, a child is 14 times more likely to die before attaining the age of 5. That is because of the effect of protein and energy malnutrition. So there's a need to increase the demand for alternative inexpensive protein sources with balance in indispensable amino acids, and those that are high in digestibility, such as the snail meat. However, in Kenya, because of cultural issues, snails are not per se consumed. So promoting consumption of these can be done through the use of alternative compositing, like the use of bands. So this study was set out to develop the sorghum wheat bands, which are composited with the snail meat powder, and we used a rat bioassay to assess the digestibility levels. The next slide. The materials, bands were formulated. So the procedure that was used was according to what Ayo and Kama had used, so that we could have 70-30 ratio of the cereals, that is sorghum and wheat. And we prepared 6 different types of bands, from 0 to 25% compositing with snail meat powder. And then that's for the rats. That is when we are now evaluating the bands. These diets were prepared according to AOAC international procedures, method 960.48 with modification. So the modification was based on the protein levels of 8%, because the 0% snail, the 0% sorghum wheat bran had a protein percentage of 9.93. Next slide. So the next slide, that is table 1, it's showing the approximate composition of the bands that were used to formulate those diets. Move on. Then table 2 now shows the formulation of those diets so that we could make 1,000 grams for the diets. That is those diets that are 0% snail meat powder, 5, 10, 15, 20, 25, and then the reference diet and the basal diet. Then a rehabilitation diet was also formulated. The rehabilitation diet had 20% protein, so it was formulated to get to 2,000 grams. Next. So the animals that were used were the rats. So we had 32 rats, the weanling albino rats. They were aged between 25 and 28 days old, with an initial weight of 37 grams and 41 grams in that given study. And the rat bioassay study followed the procedures of the AOAC International method 960.48. Next. So the first thing that we carried out was the growth study to see if these diets could be able to facilitate growth. And the growth studies were conducted between a period of 28 days with an initial 3 days acclimatization. Next. Then out of this growth study. So the protein efficiency ratio, food efficiency ratio, net protein retention studies were carried out, and these ones lasted for 10 days because those rats that were fed on the basal diet were losing weight, and they could not be allowed to lose more than 20% of their initial weight. Continue. Then what followed was the rehabilitation studies for the rats that were on the basal diet because they'd already lost weight after the 10th day. So from the 11th day, they were put on a rehabilitation diet so that they could not continue being demarcated. So the rats that were fed on the basal diet, having recorded that, they were now to be put on a 20% protein diet. Continue. Then we also did an in vivo digestibility and protein quality evaluation. The digestibility study lasted for 5 days. It was within the 28 days of the growth study. So between the 5th and the 9th day, then we were recording the food that the rats were able to consume on each day vis-a-vis what remained at the end of the day so that we could know how much they've consumed for the day. And then the total fecal material was also recorded. So we collected the fecal material, and these ones are basically used to analyze the levels of protein digestibility that these rats were able to consume within that period of time. The fecal nitrogen was determined using microjedal method. Then the fecal material from each group of rats was pulled together and was used for that specific study. Next. We also assessed the protein quality using a protein digestibility corrected amino acid score and the digestibility indispensable amino acid score. The PDCAS, that is protein digestibility corrected amino acid score, was basically to determine the indispensable amino acids that were in the diet in terms of the requirements for children aged between 3 and 10 years. And then the digestibility indispensable amino acid, this one was was to consider the regulatory purposes of this protein for that very age. Next. So data was analyzed based on mean values and standard deviations and were presented in tables and charts. Data was analyzed using one-way analysis of variance with SAS version 9.1, and the least significant differences were determined at a p-value of 0.05. Next. Now, in terms of the results, the growth and rehabilitation studies, as you can see from the table, the protein efficiency ratio increased as the fortification levels increased, the control diet had the highest protein efficiency ratio. The 25% fortified snail meat bun diet also had almost levels equal to the control diet. In normal circumstances, those diets that normally have the protein efficiency ratios of not less than 2.1 are normally believed to be complementary, and they are normally recommended to promote growth in children. Then the food efficiency ratio also increased with the level of fortification. As you can see, the control diet had similar food efficiency ratio to the snail meat buns fortified with 25% snail meat powder, and this one is believed to enhance basically the food efficiency ratio of the fortified buns was close to the reference as compared to the unfortified diet. So the low food efficiency ratio of the unfortified sorghum wheat bran diet probably led to it had low quality of protein in it, and that's why the food efficiency was low in the 0% snail meat powder bands. Then the net protein retention ratio, was also higher in the control, and it was similar to the 25% snail meat fortified bands. The net protein retention is basically, when you look at this specific growth study, when the net protein retention ratio is higher, it normally indicates the benefit of growth in terms of having an improved growth of the experimental animals. And this one can also be seen on the body weight, because the body weight also increased as that given result. The rats that were fed on the basal diet actually lost weight, as you can see on that given table. Next. So on this graph indicates the mean weight increase in rats that were fed on unfortified sorghum wheat bran diet and those fed on sorghum wheat bran diets within a period of 28 days. The red line, the lower line, the red one, that one is for those rats that were fed on the basal diet. So on the first 10 days, you can be able to see that the graph is going down, meaning that the basal diet did not support an increase in weight. But when now the rehabilitation diet came in, so as you can see from day 12, there's an increase of that. The graph is going higher, meaning that the rehabilitation diet was able to enhance weight gain and growth in those rats that were initially on the basal diet. So that one basically indicates that if a child is fed on diets that have low protein quality, then this child is going to lose weight. But if given a diet that has higher protein, then basically the same thing that happened with these rats will happen to that given child. So they'll be able to gain weight along. If the study was to continue longer, then probably this graph would have matched the other one on the others on the top based on the protein quality. Next. Then on in vivo protein digestibility, there was a significantly higher food and protein intake in rats fed on sorghum wheat bran diet fortified with snail meat powder and the reference diet as compared to the unfortified sorghum wheat bran diet. And this was perhaps due to an enhanced quality of the indispensable amino acids in this given diet. The food intake increased as the fortification levels increased. The protein intake also increased in a similar fashion. Then if you can look at the fecal output, the fecal output was higher at 0%, meaning that there was less of the food that was being utilized in the bodies of these rats. That diet was at low protein quality, so less of the diet was being utilized, and that's why the fecal weight was higher. Then the fecal protein output was also higher when the diet was not fortified, and the protein retention ratios increase with the increased fortification. As you increase the amount of snail meat powder on the sorghum wheat bands, again, the protein retention ratios also increased. So we can say that a reduction in food and a protein intake of rats fed on the basal diet might have resulted from a deficiency of the indispensable amino acids. And of course, there's a possibility that also maybe this kind of a diet has low vitamins and minerals, and actually it normally leads to an imbalance in nutrients. Next. Then on protein nutritional quality, we consider the apparent protein digestibility and true protein digestibility. So the apparent protein digestibility values of the reference diet, that is the control diet, and that of the sorghum wheat bran fortified diets was significantly higher. And as you can see from the table, from 5% going up to the control, there were 86% and higher values of the apparent protein digestibility. So it may be, it's believed that a higher protein, apparent protein digestibility normally led to the low fecal output, as we have seen in the previous table, and considerably more digestible protein. So it's an indication that if you increase the fortification levels, then we are improving the protein source in that given diet, as well as the digestibility levels. Then the true protein digestibility of the unfortified sorghum wheat bran diets was low. If you compare this to the reference diet and also the other sorghum wheat bran fortified diets, so the increasing true protein digestibility is an indication that there was an improved protein quality that enhanced the digestibility of foods, and that's what led to an increase in the values there. In the next table.
I think
Yeah.
your
Yes.
time is up, but try to summarize, please.
Okay, let me summarize. So the next table is on the PDCAS. PDCAS normally tells us which proteins are available in a given food in higher quantities. So we normally use the true protein digestibility to assess. So in this diet, the limiting amino acid was the lysine. Next. Then the next table is table 7 is on digestibility indispensable amino acid score. And this one is normally an indication of proteins that are normally, diets that are normally good for regulatory purposes. And if you can look at what we have, again, these diets were limiting in amino acid, lysine. And actually for the diets, what was happening is that they did not meet the 75% requirement that is normally required by, it's normally considered by the Food and Agriculture Organization. So basically it means that if we could increase the fortification level up to maybe 30%, then we'll be able to achieve that. So, but all in all, the diets had at least an improvement in the lysine score. Next. So in conclusion, we can say that if you compost diets with snail meat powder, they'll promote significant growth in rats, improve net protein retention, protein efficiency ratio, true protein digestibility, PDCAS, to levels that are considered potential and can be used as complementary or rehabilitation diet. So meaning that this given diet, if given to children, we expect to see an improvement in their growth and health. Therefore, this will help in alleviating the issues of protein and energy malnutrition in young children who normally depend mainly on cereal-based diets. The diets formulated in the present study did not meet the lowest DIAS value of 75%, suggested to a certain quality of protein in that, meaning that you cannot use this specific diet as a sole food, but you must be able to complement it with other diets. That's what basically it means. So future studies should focus on enhancing fortification to above 25% with snail meat fodder for foods to be utilized as solitary protein sources in order to meet the indispensable amino acid requirements for children aged 6 months to 3 years. Thank you.
Thank you very much for your presentation.
Thank you so
I
much.
don't know what ARAT bioas is.
Bioasset, yes.
Bioasset.
Yeah.
I was wondering. And so maybe is there, if you have any question, we are running out of time, but you can give you maybe 1 or 2 questions. Is there anyone with a question, please? Or those who are participating in this room? Anyone can lift their hand or write something on the chat. Okay, let me just ask you one or two.
Yes, please.
Why snail and not groundnuts? You know, snail already creates nightmares. So
Yeah,
maybe
sure.
I'll ask you that.
Yes,
Then let me ask you
yeah.
the next one.
Yes, please.
I've seen that your control seems to have better figures.
Yes.
Whatever those protein efficiency
Yes.
than your highest
Value.
snail, highest value of snail meat certification. Maybe you can comment on that.
Yes.
And then I was also curious on what was in the desert diet that almost killed the rats. And
Okay.
also what was in the control, because it seems to be very good.
Yeah.
And also the last one is after you have proved that this can work, when will it get to our tables, you know?
Okay. Okay, let me start with why the snails. Actually, currently because of the changing climatical conditions, we can no longer depend on the conventional animal sources as protein sources. So it is good for us to go out of our way and look at other animal sources, the viable one that we can have. So snail is a mini livestock that we can be able to get. It is consumable in other parts of the world. It's only that it is not consumed in Kenya. And that's why we could not, in the study, we did not just use snail the way it is, but we were able to put it into another product. So the main concern is because of the changing times vis-a-vis the requirements, the health requirements. So we are looking at more animal protein sources. So snail is one of the animals that you can be able to venture in. And number two, the control was made of skimmed milk powder. And skimmed milk powder is a higher source protein. That's why you can be able to see that it had higher values than the 25% fortified snail meat ban. So that's basically the thing. Then the best diet was protein-free diet. It did not have any protein in it. The protein was zero. Yes, I think those are the questions.
You have not answered the last one. What is the next step now that you have found all this?
Okay.
When are we going to consume it?
The next, actually, the next step is we are trying to have this on the table. We did a sensory study actually with the children and adult consumers, and they gave us their result. Actually, our paper has been accepted. It was accepted yesterday. So we believe that because of that, since people can be able to consume this kind of a product, we need to ensure that we put it on the table so that people can have it.
Thank you very much. I'm not seeing any question on the chat. Maybe you want to ask a question, but I'm also not seeing the other presenters who have gone. I think if anyone has a question or a comment, it's now your time as we close.
From my side, I don't have any, just to say thank you. Thank you for this opportunity.
Okay, the others also look to be okay. So we end our presentations today with that. And I think I wish each one of us a happy hunting as you go looking for good research things to research topics to bring to us so that we can continue to make a difference in food security. Thank you very much.
Thank you so much. Appreciate it.
Maybe we can say bye. Bye-bye. Bye.
Bye. See you.